Good morning, good afternoon, good evening, wherever you're joining us from in different parts of the world. Welcome to the second day of the Global Young Scientist Summit. This year we celebrate the 10th anniversary of the GYSS, and I think it is a very right time that we start to think about how do we prepare for the next pandemic. Now, the world has seen the problems of COVID-19 since the start of 2020. But I always highlighted that the response that scientists, the global scientific community has mounted has been one that was staggering and immensely impressive. If we just look at 4 points of innovations that have happened, from the antigen rapid test kits or the lateral flow test kits, which is what's called in some countries, where you typically transform a diagnostic laboratory into a piece of innovations that you and I can test ourselves in the comfort of our home within 15 minutes to know whether we've been infected with COVID-19. We look at how digital contact tracing has really shifted the way that we tackle the spread and understanding how do we isolate and quarantine people as quickly as possible. Vaccines, definitely within a very short time, in less than 12 months, we see the design, the clinical trials being put in place to really push forward in a very quick manner, but in a safe manner as well. Vaccines that are able to really prevent the worst of a COVID-19 infection. And finally, in terms of communications, how the digital communications have really shifted the way that governments, communities talk to each other, share information with each other on how to prevent, how to protect themselves, and also what are the measures that governments are introducing. But of course, communications is a double-edged sword. We have seen how this has also led to the spread of misinformation and fake news. Now, when we start to think about 2021, which was just last year, about this time, I actually received my first dose of an mRNA vaccine. And that was when I was filled with hope that 2021 would actually be the year that we say goodbye to COVID-19. But of course, we have seen the realities. Looking back is easy, but just about 6 weeks back, the world was faced with a new variant, the Omicron variant, that ravaged the world and has really transmitted and spread very quickly to different parts of the world. So what will 2022 hold in terms of COVID-19? And more importantly, how will the world prepare itself to handle the next pandemic? Or sometimes I like to say, we don't have to wait for the next pandemic, but perhaps the next coronavirus variant. So today, this morning, it's my privilege to be able to moderate the session with 3 very eminent and very distinguished scientists. Professor Thomas Cech, who is the Nobel Prize winner in chemistry in 1989, Professor Robert Langer, who is the Millennium Technology Prize winner in 2008, and a dear friend of mine, Professor Wang Linfa, who oversees the Emerging Infectious Disease Program at Duke NUS and is one of the co-leads at the World Health Organization R&D, the research and development platforms, specifically looking at animal origins and epidemiology of the diseases. So I always like to highlight that the GYSS, it's never about us, never about the moderator, never about the panelists. It's about how young scientists, like many of you who are tuning in from different parts of the world, learn from some of these scientific giants. And one day, just as I hope I will be able to, one day we hope to be able to stand on the shoulders of these scientific giants to really progress and contribute what we have done in our own research. So today, this morning, I urge you, young scientists, to really make use of this platform to ask questions. And we will actually like to invite you online as well. And that's when you could pose your question to some of these eminent scientists that we have joined us this morning. But I thought before we start, I would also like to invite each of my eminent panelists to share a little bit about their thoughts of how they see COVID-19, as well as this theme of preparing for the next pandemic. So first off, maybe I can invite Bob, Professor Robert Langer, for your quick interventions. Over to you, please.
Sure. Well, I wish I had some crystal ball, and I'm not a great predictor, but I do feel that science is just, is one thing that's very important. I think, obviously, in this case, political issues are incredibly important too. But on the science issues, I think you addressed a number of important things. I think the vaccines, that's something I've been personally involved in. And really, so much of this comes from basic science. I mean, the success of some of these vaccines, we've been involved in actually creating nanoparticles that could deliver large molecules back since the mid 1970s, when people really felt that wasn't something that could be scientifically done. But now many groups have done that, and I think that's led to some of these mRNA vaccines. That coupled with great work in molecular biology and chemistry in terms of modifying messenger RNA. So I think we have but even that you can't do overnight. I mean, you could design it overnight, but you can't manufacture enough to treat billions. I think there's a lot of other areas you mentioned. The diagnostics and contact tracing, and I think that's really important. I personally feel one underrated area has been masks. I think that that's something, sadly, that a lot of both governments and the World Health Organization really recommended against for quite a period of time. And to me, masks are such an easy thing, but I don't think there's been, like what I'd say, an Operation Warp Speed on masks, and I always thought that would be a useful thing. I mean, to make better masks, safer masks, maybe even better looking masks, so people would wear them, more. So I feel like that's been something that can certainly be done to help in these situations. So I think science, and I do feel when I look at what's happened, and I believe for the future, science plays a critical role. I think the unfortunate thing has been, that some of the government leaderships, including our own, some of the world leaderships, I think that there's just so many aspects of non-scientific things that are also so important, and I feel we need to do better on.
Thank you very much, Bob. And definitely some of the pertinent issues that you talk about, the importance of non-pharmaceutical interventions, because really when faced with an emerging infectious disease, how you actually tackle it from a non-pharmaceutical perspective is of utmost importance, because it takes time to develop pharmacological, therapeutics, and vaccine solutions to address a problem like an emerging infectious disease. So the ability to adopt very simple measures like what we talk about, the mask, actually it's quite important. But that ties in with some of the issues that you talk about around this disconnect between the scientific response and a political response. Later on, I think we'll do a deeper dive in this. But next on, I would like to invite Tom for some of your thoughts before we start. Tom, over to you, please.
Well, thank you, YY. I'm just going to add on to what Bob Langer said. Certainly, the scientists and engineers have many abilities to help out with the technology for the next pandemic. Even if it's not a coronavirus pandemic, I think that surveillance activities at major airports and ports based on PCR to multiple classes of viruses, to look for newly arising viruses, maybe figuring out how to get mRNA vaccines pre-approved for use without a long FDA approval, because if the lipid nanoparticle is safe, if the mRNA modifications are safe, if the mRNA itself is safe, does it really matter which viral protein is being made? I think that's an open question. I'm not claiming that I know the answer, but I think it is worth considering, because any of the young students in my laboratory could create a new vaccine sequence and express it in a week. That's how quick this mRNA technology is. So it's then all of the manufacturing and the safety studies and the approval that takes time. And also antivirals could have could be developed much better than the few that we have. Many of these viruses are RNA viruses. That happens to be something that I have studied, is ribonucleic acid. And, the RNA-dependent RNA polymerase is a unique animal to these viruses, not so much found in our own bodies. It's just not present in humans unless we're infected. And so this is a good target. And many of these antivirals are pan-viral. They will hit everything from Ebola to coronaviruses to cold viruses. So, but also, as Bob said, the problems really that we as scientists and engineers have done really badly with are the social science and communications aspects. In the United States and in many other countries, we have seen that only a portion of the population is convinced that they should follow the science. And the approach that governments have taken is to belittle these people, to chastise them, to plead with them, to mandate that they get in line. And those approaches turned out to be naive, unsuccessful, and in fact polarized the population. So I think that we need to learn more about how to deal with these pandemics from a humanitarian point of view rather than just from a science and engineering viewpoint.
Thank you very much, Tom. And that really is something that we have been seeing quite significantly in our part of the world, where there's always this conversation around whether the Eastern culture response have differed dramatically from what is loosely defined as the Western culture. But really, this bit about the behavioral and the social sciences have really highlighted the importance that when we address a problem, a crisis, actually it's not just what we can design in terms of technology, in terms of medical solutions, but equally, do we understand the human behavior in terms of their willingness to take on vaccines, their willingness to take on masks, like what Bob mentioned earlier on, to wear the mask when they're moving about in public. This social sciences or implementation sciences actually need to be incorporated in many aspects of policy making as well. So I think we'll come back to that as well. But equally, Tom, maybe I could give you a heads up. One of the possible questions that will be coming our way is really this issue around the safety of mRNA vaccines, because we've seen there has been a lot of concerns around the mRNA vaccines in the initial phase, because I believe the world very quickly realized that it does help to be vaccinated against COVID-19. But just last week, we saw an announcement that perhaps it will not be that safe to keep taking repeated booster shots of the mRNA vaccine. So perhaps later on, when we have the opportunity, I'll invite you to share a little bit of this, given that you are the foremost expert in RNA technology. Now, next, over to Linfa, very quickly for some of your thoughts. And I know you're going to share with us some of your concerns, your deep concerns.
Thank you, YY. Yeah, so I think, I agree with what Bob and Tom have said. So I'm going to just come from a slight different angle, my personal perspective. I think I count myself fortunate because I have been, in the last 30 years, I've been involved in really investigation outbreak response for the 6 major emerging zoonotic sort of viruses of bat origin. So I started with Hendra virus in Australia and then the Nipah in Malaysia, Singapore. But I'm going to zoom in onto SARS-1, right? So SARS-1 started 2002, 2003. In terms of science, we were not ready. We did not have mRNA, the rapid test, and so on. So lucky that COVID-19 happened in 2019. If it's in 2002, it could be much worse. But the sad thing is that after the SARS outbreak, it's still a rapid spread and 8,000 people, infected, a 10% case mortality. And it was really complete control by lockdown, quarantine. And the scientific community, the international community, and the scientific funding body basically did not take this seriously. Like most of the research stopped and not enough funding. And then we wait until 2012, and we had the MERS coronavirus, from Middle East. And even then, I was predicting that every almost 10 years, there will be another emerging kilovirus from bats. And yet, still, I think, some international funding body, the scientific community, the political leaders, nobody took this seriously. So I think the lesson is there. The question is, can we learn now after COVID-19? So I'm going to propose, I use the concept a lot in pandemic preparedness, just like fighting a military war. You have the peacetime and the wartime. And, we're talking about the tsunami right now, in Tonga. And so I think in terms of these big impact events, you can have a military war, you can have a natural disaster, earthquake or tsunami, and then you can have a pandemic, okay? Unfortunately, the military war is the easiest to prevent, and yet every government spend much more money on the military preparedness than pandemic preparedness. So I think, personally, I think this is a time to waken up. This is a global threat. Pandemic sort of human health, human lives, economic loss. COVID-19 just demonstrated it's much more powerful to really disrupt the society and take lives away. So I think that this is a time, everybody and scientists work with, policymakers. I think that this needs to be a long-term strategy. I think, if we can copy the funding, training, collaboration model of military preparedness, then I think at least, COVID-19 made a positive impact on the society. Yeah, so I just stop there. Thank you.
Thanks very much, Linfa. And I think you highlighted this bit about learning from previous pandemics and outbreaks. And I think some of the jurisdictions that have done particularly well in managing COVID-19 especially in the early days, jurisdiction including Hong Kong, Taiwan, Singapore, we learned our lessons painfully from SARS back in 2003, where there were quite a number of deaths, particularly happening within the hospital sector. Even for South Korea, they amended some of their infectious disease laws around privacy and around the use of personal data because of their experience previously with MERS, I believe in 2018. So really this bit about learning from previous outbreaks is a very important aspect, particularly from a government response. And I believe that moving forward, there will be a lot more efforts focused on really preventing and avoiding future emerging infections. And that's really where some of these discussions around investments, around training, around infrastructural development will all have to come in. And also, very importantly, about equity around the world. So these are some of the issues that have been our eminent panelists have shared. So again, at this point, I will issue a call to all our participating young scientists, make use of this opportunity to ask the questions to some of our panelist members. And in a while, I would actually invite some of our young scientists to join us to pose the questions. And if I can handle the technology well enough. So particularly Lawrence Tan, I will invite you in a bit to share your question and to ask your question yourself. But before that, I wanted to come back and pick on what Bob, you have mentioned earlier on. Really, some of the disappointment that you felt in terms of the disconnect between the scientific evidence and the government response, because we have seen in many countries, particularly in advanced economies, which is what I always like to highlight. These are the countries that housed the top brains in science, in epidemiology, in medical sciences, in clinical sciences. And yet many of these countries, the government, the political response have been one that was disappointing and that was quite backward looking, particularly in the start of the pandemic in 2020. So Bob, in your opinion, how do you think that as scientists, you and I and others, we can influence this process of policy making? Because I believe for too long, scientists have remained in the comfort zone of our laboratories, of our computer server rooms, without really engaging the real world on the policy side. So many of you here have actually made the contributions to impact lives in a real world manner, but there are still equally many epidemiologists, many researchers that are very much focused on research per se, but not really thinking of making that translation from research to really impacting lives. So what are some of the recommendations and your thoughts on this? Bob, over to you.
Yeah, well, it's a great question for which there's not a simple answer. I mean, I'd like to think I'm an okay engineer, but I'm not probably a great politician. And I think that may be true of a lot of scientists. I think two things that end up influencing public opinion are the press and obviously politicians. In that sense, it would certainly, to me, be better if there were some scientists who were at a high level in politics. But that's not a simple thing to do. And I don't know, you have to have a certain type of personality, I think, to do it well. Myself, I've done things like I've been chair of the FDA Science Board. That was their highest advisory board, and I've certainly been involved, and I'm sure Tom has, I know as well, in different things. But those are part-time positions, so people, you give advice and you do the best you can. But the people with the full-time positions, are the ones that do things. And then there's the press, which I think has been really disappointing. And a lot of people, obviously, I mean, I don't want to overgeneralize, but there's been an awful lot of inaccurate misinformation. I think Tom pointed to some of that, whether it's the press or various, people twittering and things like that. And that's not that easy, in a society, to change that. So I guess my recommendation would be to have more scientists get involved in both the media and in public policy. But you have to recognize when you do that once people start doing that, they're probably going to be less involved in science because it's very hard to do everything.
That's definitely true, because Linfa has been one of the very vocal and very helpful voice to really share his knowledge with the public in Singapore and beyond on some of the developments. But I expect that some of the time that he spent with the media, with the press, as well as guiding some of the WHO movements, have definitely taken a lot of his time from his own research. So, Lawrence, may we invite you to come on screen to pose your question, which is really around some of the issues and concerns on the speed of vaccines and therapeutics approval. Lawrence, over to you. Are you able to join us on screen?
Hello.
Hi, Lawrence. We can see you.
Can you hear me?
Welcome.
Hi there. Brilliant. Thank you very much for the opportunity to ask this question. So basically, I was just thinking, with the speed of the delivery of the therapeutics that we've seen and the vaccine approval throughout the pandemic, do you think that there needs to be a regulatory overhaul to remove any unnecessary bureaucracy going forward?
Thanks very much, Lawrence. So your question on the speed of therapeutic and vaccine approvals, that both Tom and actually Bob have mentioned that it is typically a very conservative and a much slower procedure to really approve some of these developments. Should there be a regulatory overhaul to remove some of this unnecessary bureaucracy? And perhaps, Tom, earlier on you talk about the speed being a lot slower, and in fact we could actually prepare in advance, particularly some of this, your thoughts on the antivirals. What are your thoughts in terms of this regulatory approval or regulatory overhaul to try and really minimize some of the red tapes in approving tried and tested products, or perhaps even some of the untested products? Tom, over to you.
It of course is a very it's a heavy question, a weighty question, because the thing about vaccines is that they are given, first of all, to very large numbers of people And secondly, they're given to healthy people, including children. So the sort of low incidence of adverse effects that for a cancer drug, you would say, not a problem, because people are going to pass away if they don't get treatment. So if they have a 1% chance of an adverse event from this medicine, or even a 5% chance, that's quite tolerable. A 1% chance of adverse events for a vaccine would be completely unethical and unacceptable. So that's where we're between this rock and a hard place. As an RNA scientist, I really have a strong feeling that that once we've approved the Moderna and the BioNTech Pfizer mRNA vaccines, that the next one shouldn't have to go through such a lengthy safety study. However, I must admit that, you know, we need to be safe, and if we're not safe, then the anti-vaxxers will have a tremendous club to wave around and hit people with. So I just think there needs to be more thought given to this issue, but I don't think there's an easy answer to it.
Thank you very much, Tom. And at this point in time, I think our theme has really been talking about preparing for the next pandemic. And I'm going to pose a question to Linfa. But while I'm doing that, I would like to invite Nina Alyssa Baroga to join us on screen to ask your questions really around boosters. So Nina, just a heads up that I will be coming to you after Professor Wang Linfa has a chance to speak. So Linfa, I wanted to come back to you and really pick up on something that you mentioned earlier on. So far, the theme for this session is really preparing for the next pandemic. And we've been thinking along the lines of how scientists and governments should react when a pandemic actually happens.
Yeah.
So it's responsive. But how do we switch from a responsive approach to one that is more proactive? And what are the preparations and recommendations that you think we should put in place such that we can truly be prepared for the next pandemic? Over to you, Linfa.
Thank you, YY. I mean, I would say it's a non-million-dollar question. It's a billion-dollar question. It's very important. But when you think of pandemics, right, you know, not exclusively, but most likely the next one will be a zoonotic one. So if it's already a human pathogen circulating, suddenly become very virulent and become a pandemic, the chance is low. So you have a pathogen circulating in animal, either we know or we don't know, and they suddenly spill over to human and slowly or rapidly cause a spread. So in terms of to mitigate that, you really have three different what we call stages or levels. The first level is we call pre-spillover. So when the animal is sort of starting to have this noise chatter, as we say, the virus is kind of jumping around to other animals or to humans, but distinguish very quickly. So this is the pre kind of spillover before they establish in human infection. So that, you know, I mean, Tom already talking about the active surveillance. So that one I think is important, but it's really, really difficult because there's so many viruses, so many animal species all around the world, and where you target. The next best thing you can do is what I call early warning and mitigation. Okay, 2019, if the world act together in Wuhan, transparently work together, the first 3 weeks of December 2019, you know, if the best brand, all the money, all the technology was really sort of focused on Wuhan, we may not be here today. So that to me is the most critical. And then past that, then it's what we call, you know, post-outbreak, then the countermeasures, the vaccines, the therapeutics. So I think, you know, both Bob and Tom already touched that. So I think personally, in 30 years of pandemic outbreak response, I personally think, if you want to have, the big bang for your investment money, I think it's in the early warning and the mitigation. Basically that, but to do that, then I think science, I always say, is only 50%. The other 50% is international collaboration and the legal framework. WHO always play a very important role, but legally, WHO don't have that right to send a mission to the outbreak site, for example. So in a different forum, I propose the concept, maybe WHO plus Interpol, In terms of civilian crime, we have an Interpol, the international collaboration. It's the common enemy. And for virus, why can't we do that? Politics complete out, just transparency, collaboration, and, then the science, can really play a role, because I think the science is so advanced, although we can improve, but I think if we put all the scientific, brain and without the politics and without the legal frame hindrance, so you talk about, the question about the regulatory thing for vaccines. I think that the policy during peacetime should go beyond the regulatory for vaccine therapeutics, sample handling, biocontainment safety. We have every SOP established for the lab to do SARS and MERS, and yet when COVID-19 emerged, the virus is almost identical to SARS, 80% identical. Every time we have to repeat the paperwork. So all of this, I think, has to really, have a new think about it, because if we use the current, legal framework, political framework, and regulatory framework, I think the science alone really cannot do much better to prepare for the next pandemic. Thank you.
Thank you very much, Linfa. And at this point in time, I actually wanted to also introduce that Linfa, with his knowledge, has actually been appointed as the executive director for this initiative or this program that we have in Singapore that is called Prepare, specifically with the intention to prepare for the next pandemic. So I like the fact that Linfa actually broke it down into really 3 different forms of responses, the preparations that happen, the preparations that you need to put in place before any outbreak actually happened. And that's really how are you monitoring, how are you surveying the animal kingdom, the host and vector interactions. The second part is really around the first few days or perhaps the first few weeks, if you're lucky, of how you respond very quickly to evidence of an outbreak. And then the third is what Linfa talked about, really is the pandemic response that we are starting to see in terms of therapeutics, the use of non-pharmaceutical interventions, or perhaps I can add on a 4th response, which is the endemic response, where we realize that vaccination and preparing the healthcare sector is likely to be a response that governments need to put in place. So thanks very much, Linfa, for some of your thoughts on this. And at this point in time, I want to come back to Nina. Are you able to join us on the screen? If I could invite you to turn on your camera. Nina, please pose your question, please.
Hi, good morning. Can you hear me?
Yes, we can.
Yes.
Loud and clear.
I am Nina. I'm actually from the Philippines, but I am a PhD student here in Academia Sinica, Taiwan. So I think we already know that Taiwan has been really controlling the pandemic successfully for the past 2 years. Actually, just last year, we already just had the peak of COVID, around 600 cases only per day, and that's really just our peak already. But however, despite that, there are still some countries that still having the outbreaks and surges. So it seems that global herd immunity from this pandemic is still far from reality to achieve. So do you think people will still continuously receive booster shots? Because I think it would be tiring that maybe we just have to live with it. The COVID, we live normally with that. So what's your take on this? Thank you.
Thank you very much, Nina. This question on repeated booster shots and perhaps including even the safety of repeated booster shots, it's definitely of great concern these days, especially given last week's announcement that it may not be safe to be taking repeated shots. Bob and Tom, I would like to invite both of you to comment on this. Perhaps Tom first, and then Bob second. Tom, over to you. Your thoughts.
Well, I think your first question, Nina, was about whether we really should be giving booster shots at all. And I think that there's a breadth of people in the population that have different susceptibility to an adverse event if they get the virus, right? And so there are people who are young and do not have any underlying medical condition, who appear to be just getting the virus and often don't even know it, right? And so for them to get a booster is more questionable. However, for the people who are have underlying medical conditions, are immunocompromised, or are much older, often the outcome from a viral infection is not such a happy one. And it's been shown that the booster of the mRNA vaccines makes an enormous increase in both antibody production and cellular immune response, really increases the protectiveness a lot. So I think it would be premature to say that we should just not be giving boosters. I think there definitely is a place for the boosters. I don't know, Bob?
Yeah, well, I completely agree with you, Tom. I mean, and I'll maybe just say an additional point. I mean, right now, at least in the United States, I mean, most people that certainly that are older, but even younger people sometimes are recommended to get a flu shot every year. And what they do with the flu shot, I mean, the flu is, each year it could be a little bit different, like H1N1, whatever. And so but every year, I mean, I know I've always gotten a flu shot. And by the way, I think the important thing, flu shots haven't even been that effective. They're less than 50% effective, whereas the Pfizer and Moderna, shots are 95% effective. So I think so my feeling is that, the term booster probably needs to be defined. I mean, it may be that every year, depending on the mutations or whatever happens, I don't think we know what's going to happen in the future, whether you're going to keep using the same basic injection or whether you'll use something aimed at a slightly different mutation of the spike protein or whatever. But I think what Tom said is absolutely right. I think that, just as we do with the flu, taken every year, at least a significant population of the world, I would think for something that's much more lethal than the flu, which COVID has been, that you would do that.
So thanks, Tom and Bob, for your thoughts on this. But if I may, I would like to push you a little bit on the safety then, because there have been a lot of concerns when the mRNA vaccines was first introduced. And I believe this concern has been somewhat allayed now, given that we have delivered billions of shots of the mRNA vaccines globally. But now there's this concern, is the human body able to take sustained and repeated exposure to the mRNA vaccines in terms of the safety profile of this mRNA vaccines? Or should we now switch to more traditional vaccines methods, such as what has been used in the influenza vaccines? Tom, any thoughts on this, please?
I am not an expert vaccinologist, and so I think that there's quite a bit of data coming out of Israel where they've given many people a third shot and are now giving a fourth shot. And I don't think that there are enormous instances of adverse events. I mean, the best medicine to take is none at all, right? That's if you ask the people at Merck, where I used to be on the board, they would say the best drug is don't take any drug, right? So, but sometimes, a pandemic happens and that changes the risk-benefit ratio. So I will say that many of these adverse events that are very rare with the mRNA vaccines are similar to what one would get at a much higher frequency if you got the disease itself. And that is typical of vaccines, that the adverse events are not just something from the planet Mars, but they are something that people who get that disease experience at a much higher frequency. So I think a lot of people don't understand that.
Thanks very much for that reassuring note, Tom. So we have a question that was posed by two members of the audience, and I would invite you both because it's along the same vein, but if I may, I will read out your names, Yeo Yai, as well as Machima Monkan Rata Nachai. Both of them have asked very similar questions, which I would like to direct to Linfa. And this is really around the variance, the diversity of viruses that we see in the animal kingdom, and whether there is even the possibility of predicting this variance of viruses better using computational experimental models, techniques that are already being used, very common in bioinformatics. Linfa, what are your thoughts to this?
Okay, so I think that there's two different layers, right? If you talk about virus in general, so for example, I have been studying bat-borne virus for the last 30 years, and there are many, many viruses there, and the coronavirus seem to have a special affinity with bats, so there are many of them. So at that level, if you just go de novely and survey through all the data bank and try to pick up the virus, let's say from a rodent or bats, and try to make a prediction, say which one is ready to jump, I think that's still a science fiction. I think, we're not that advanced because, even with a small virus of, 10,000 nucleotide, you still have 10,000 nucleotide. Even one single nucleotide change can change the phenotype. And for coronavirus, we have 30,000 nucleotide. So at that level, I don't think that we're ready. But now specifically come to SARS-CoV-2 variants. Okay, so I mean the ones that the public knows, the alpha, the beta, gamma, delta, and Omicron now. But if you look through the data bank, if you define variants as a genetic change from the original Wuhan parental virus, then we have tens of thousands already. Okay, so from there, can you predict, which mutations will be more likely to escape the current vaccine? So that one, I think we're getting closer. Not 100% accuracy yet. And, so Omicron, for example, has 30-something mutations even in the spike protein. And then the receptor binding domain, I always say the receptor binding domain is like a key, opens the door to enter human cells. And within that region, we are talking about only 250 amino acids, and Omicron has 15 mutations. So first of all, I think no one would have predicted the virus evolved that fast and also have so many different mutations. But we are getting better with birth. I think the bioinformatic modeling is helpful, but alone will not really be able to predict very accurately. So now there's another kind of research called a saturated mutation analysis. Basically make changes of every residue in that domain and try to make sense. So I think that, unfortunately, the outcome is not as rosy because you cannot just use computer to predict which virus is able to jump or which variant is going to be more transmissible or more virulent. But with SARS-CoV-2, we are learning, and hopefully that knowledge can be applied in the near future.
So, Linfa, stay on the line because I have an additional question to follow up on what you have mentioned, which is posed by Fabian Franco. Fabian, I will not invite you to the screen, but I will read out your question because it's a nice follow-up. But I just want to give Sumina Singh a heads up. I will be inviting you to join us on screen in a matter of minutes after Linfa addresses Fabian's question. So Fabian, your question was really a very nice follow-up from what Linfa has mentioned. Let's zoom in specifically on SARS-CoV-2 then, that causes COVID-19.
Sure.
We have Omicron. And Fabian's question is, after Omicron, what is the likelihood that the world we'll be faced with another variant that may be more contagious and perhaps even deadlier. What are your thoughts? Because there's a lot of theories that are circulating online whether Omicron is likely to be the end game for COVID-19, or in fact, like what WHO warned about, there is the possibility of more dangerous mutations coming on the back of Omicron. Your thoughts, Linfa?
Well, first of all, I think in science, you never say never. In terms of have another major variant after Omicron, definitely possible, because even scientists like myself, we have been studying SARS and MERS for the last 20 years. I was still surprised how specific our immune responses are, right? Alpha, there's only one amino acid change in the RBD, and Delta, there were only two, and already escaped some of the vaccine efficacy and also made it much more transmissible. So the immune response is very specific to the virus strain or variant. So even with Omicron, there's certainly a possibility of another variant comes and can escape the current Omicron immunity. And that's what, I'm conflicted, because I'm trying to really promote that we need a better vaccine. And I'm personally developing what we call a pan-sarbecovirus vaccine. So all the viruses in the subgenus of sarbecovirus, they all use the ACE2 receptor. So we need a vaccine which will be what we call variant agnostic. The big pharmaceutical company initially was going to launch a vaccine against Alpha, and then Beta, and then Delta, and now they're working on Omicron. I don't know whether the Omicron vaccine will eventually be marketed, but even if it's marketed, I think you still can be vulnerable to another variant of SARS-CoV-2. So, CEPI is putting in big monies, and I'm not alone to work on this. So I think, hopefully within the next 6 months or something, we will have the next generation, the second or third generation vaccine, at least will provide a broader protection, which will be variant agnostic. So I think to get out pandemic, we need two things. One is a better vaccine, and secondly, again, international collaboration. Because if 1/3 of the population is really protected, 2/3 are not, they will keep generate variants, come to the 1/3, okay? So, the cliche says, nobody is safe until everybody is safe. I don't want to repeat that too much, but I think it's philosophically very true. Thank you.
Thanks very much, Linfa. So, Sumina, are you able to join us on the screen to pose your question around vaccine hesitancy and misinformation? So you will be a representative voice for a very common topic of questions that have been emerging on my screen. Sumina? Okay, so I think, Sumina, it's a little bit... Oh, yes, you're able to join us. Please, over to you.
Hi there. Can you hear me?
Yes, we can.
Hi, I'm Sunima Singh. I'm from Nepal. I'm right now in New York. And it's a pleasure to be here and be listening to all of distinguished scientists like you. My question is, as scientists, especially young scientists in this pandemic, what can we do to help curb vaccine hesitancy and misinformation within our own families and broadly in the general public as well? How can we speak in lay terms to explain this to the general public?
So, Bob, what are your thoughts to this? How have you been addressing some of this misinformation and fake news, perhaps even circulating in your own social networks?
Yeah, well, I mean, it's a great question, and it's really disappointing to me that enough people feel that way, and A, give misinformation, and B, others believe it. I mean, I just try to go to the data, and try to explain it in a simple way. Just as an example, which I mentioned before, you could compare what's happening with these vaccines to what's happened with flu vaccines. If you really look at the data, and like I say, just like Tom, I'm not a vaccinologist or an immunologist, but the way I look at it is lots of people take flu vaccine, at least it seems to me that way, and they're not mRNA vaccines. The mRNA vaccines are, when you look at the data, orders of, just tremendously more effective. I mean, 95% effective. You can also have the ability to make them relatively quickly, to respond to pandemics. So one, they're more effective. Two, I realize that people have mentioned that there's safety issues. As far as I've heard from people who are experts in vaccines, those safety issues are no greater with these vaccines than they are with flu vaccines. So to me, comparing this to what happens with flu, I mean, the unfortunate thing with the COVID issue is it's much, much more lethal than flu. Maybe not more lethal than the 1918 epidemic, but really, to me, I use that as a reference point for almost everything. And I think when you do that, I'd like to hope that at least some people get convinced by that. But there's enormous amounts of misinformation and by all kinds of people. But to me, that seems like a very legitimate argument. Even what was said before, about mRNA vaccines for the fourth time, which I think Tom addressed very well. But again, people take flu vaccines. I realize they're different, H1N1, H1N2, whatever. But so what if you take four? I mean, I've probably taken, 40 or being 73, I've probably taken 40 or 50 flu vaccines. I mean, it's so what if, so I think you have to have a reference point when you say something's bad. If 1 out of a million people have a problem, I think that's unfortunate. But 2 points, that's going to happen with probably most vaccines. And Tom's point's exactly right. What are the consequences? How many millions of people have already died by not taking those vaccines?
Thanks very much, Bob. And I really like to highlight that Sumnima's question. Apologies. Yes, apologies. Your question really has been one of the very popular questions. Shireen Ng similarly asked, how can we as young scientists combat the spread of misinformation, especially in an era of social media? Tom, this is where I like to come to you, because I suspect, like myself, you would have been the recipient of numerous fake news or misinformation circulated by your social networks, perhaps on WhatsApp. How do you respond when you actually see some of this, which are blatantly false, but may come from a very dear friend or a dear relative, that it puts you on the spot to really try and correct them? Over to you, Tom.
Yes, I think we, at least in this country, the United States, we all have someone in our family or someones who are convinced that these are dangerous and that that is some kind of a political statement, and they get their information from social media, and you can always find something on the internet to support any view that you might make up. You could find other people reinforcing it. So here's Samnina, here's what I think does not work generally: to tell people that you're wrong, to tell them that you owe it to your neighbors or your family to do this, to tell them that you're an idiot, to tell them that you don't understand the science. I think that those approaches just polarize the situation. And so I think you should not, here's my most current idea. I'm not sure that it's so successful, okay? But is don't tell people what they should do at all. Just say, well, I've been studying a lot of science, and I find it very interesting the way that these mRNA vaccines work. Would you be interested in hearing about how this works? Messenger RNA is something we have in every cell in our body. It's something we have in all the food that we eat, in all of the lettuce and vegetables and meats. It all has mRNA in it. But those mRNAs are all a code that produces some protein that makes the lettuce green, for example, right? Or that makes my hair be what it is, or that makes my liver do its function, or my brain work. So this mRNA is not that new of an idea, but here's the exciting thing about it. is that it turns out that you can just type in the code for the outside surface of the SARS-CoV-2 virus, and you can make the mRNA now make something that looks like that exterior of the virus, but it's completely non-infectious. It's not a virus, it's just that little piece of the virus, right? Which by itself can't do any harm. And it's just sort of a scientific accomplishment that's as exciting as when we put people on the moon, as when the astronauts went to the moon. That's how exciting this is. And then just maybe that would make a difference to some people. But I think that just haranguing them, telling them that they're wrong or misinformed or stupid, that isn't going to help.
Thank you very much, Tom. And at this point in time, we have 4 minutes left, and I would like to invite Saki Priya Amartaraj to join us on the screen, if you could, to ask your question, because I think your question is a very nice point for us to end our conversation. If you could join us on the screen, Saki Priya. Thank you for joining us.
Hi everyone, so I'm so happy to be here and thank you for this very nice discussion. So I was just curious, I was wondering, is it possible to make an artificial virus which could, like spread faster but far less harmful, like a vaccine? But why not just make an artificial virus that can, mitigate the spread of COVID, SARS?
Oh, okay, Saki, I realized that you have posed 2 questions, because the first question that you posed about half an hour ago was actually the one that I was looking forward to. And if I could just paraphrase what you have just said, it's really around the contributions. What other contributions are required in different fields of science in order for us to prepare for the next pandemic? So you posed that question about 30 minutes ago. And I thought that was a very nice ending point that we could pose to all our panelists, really thinking beyond their own fields of expertise and looking at what other disciplines, what other thinking would be necessary, such that collectively we as scientists realize that we provide one very small piece of the jigsaw, but we do need other scientists around us. And I would like to invite all 3 panelists perhaps to address the question posed by Saki Priya. So perhaps, Linfa, I could come to you first.
Yeah, so I think we're addressing the question of different disciplines, right, contribute to pandemic preparedness, and I think we addressed the science in general already. But if you want to really zoom in, for example, early stage of the outbreak, we found that there's a huge gap on the transmission study. So this need aerodynamics, the computing scientists, environmental scientists, come in. And I think we already talked about social science and political science and the legal framework. The other big part is climate science. I have been doing backbone viruses in Australia, and the virus, Hendra virus, the bats moved from Australia is in southern hemisphere, for those of you who don't know. So we get winter in sort of June and the summer in December. So the further north you go, right, it's hotter, and the south is cooler. And so the bats have been migrating. Bats like tropical, subtropical weathers. So they have been migrating from north to south and bring the virus with it. So even the environmental, climate change have an impact on pandemics. So I just stop there.
Thank you very much, Linfa. And Bob, over to you. What are your thoughts?
Well, I think almost every branch of science and engineering is important. In the interest of time, I'll just say biology, chemistry, pretty much many branches of engineering, because you want to attack these in every kind of way. Like we talked, better diagnostics, better surveillance, new nanocarriers, new vaccines, to manufacturing and so many other things.
Thank you very much. And Tom?
Yeah, I will just add to that list computation, including artificial intelligence and machine learning.
very important additions as well. So I think it's clear that we realize that as scientists, we are specialists in our own domains. But actually, in addressing a crisis or in addressing any real world problem, we provide perhaps one piece of the puzzle, but we do need the contributions from many other fields in terms of technology, clinical sciences, the social and behavioral sciences, even the way we communicate with and engage with the media. But most importantly, I believe there is this need to stay engaged with the government, with the policymakers, such that whatever we find, we have this direct translation to policy. And then I always feel that as a scientist, I'm a mathematician in training, but I realize that I don't know many things. And it's really through that humility that we stay humble, that we know what we know, but actually in addressing the world's problems, we need the partnership of other scientists and researchers out there. So with that, I would end off this session on preparing for the next pandemic. And I do apologize that I couldn't get to all the questions that many of you have posed. But with that, I would like to end off by thanking our three speakers once again for taking that hour to join us to share their very valuable thoughts on how we prepare for the next crisis in future. Thank you again and have a good day ahead.
Thank you.
Thank you.
Bye.
Bye-bye. Thank you.
Hello, everyone. My name is Tan Chuan Singh. I'm a professor in the School of Electrical and Electronics Engineering at NTU. It is my honor today to introduce the plenary speaker, Professor Jayant Baliga, the Progress Energy Distinguished University Professor at the North Carolina State University. Professor Baliga received his BTech from IIT Madras in 1969 and his PhD from Lancashire Polytechnic Institute in 1974. And he has had a long, distinguished career at General Electric before joining NC State. His most impactful innovation was the invention, development, and commercialization of the insulated gate bipolar transistor, IGBT. He's a recipient of numerous awards, and let me highlight a few. He's a member of the National Academy of Engineering at a very young age of 45. He received the Medal of Honor, which is the highest recognition by IEEE. He received the National Medal of Technology and Invention. by then President Obama, and he was inducted into the National Inventors Hall of Fame. The topic of his plenary presentation is IGBT, from invention and commercialization to global social impact. With that, let us welcome Professor Baliga.
So thank you for that nice introduction. So in my presentation, I will talk about the IGBT device. I'll take you all the way from the invention and commercialization of the device to its enormous global social impact in this presentation. Next. So since most of you are not familiar with power semiconductor devices or power electronics, I'll start with a very simple example that happens every day in your life. When you throw a mechanical switch in your house, you turn on a power source and then the power flows and turns on a lamp. So we like to do the same thing with power electronics. So next, it shows the mechanical switch, and the next shows the electronic switch. So we can do this with an electronic switch at a much, much higher speed, at 100,000 times per second. And by doing that, we can do digital power control. And this has enabled us to get tremendous increase in the efficiency for delivering power from sources. Next. In order to achieve this, we want the power to flow from the source to the load without any of this power being consumed by the switch itself. So that is how we get a high efficiency. And in order to achieve this, the power switch has to have certain properties that are listed here. First is low on-state voltage drop so that we don't burn any power while the current flows through the switch. Next is we want high switching speed to do minimization of power losses and to improve the circuit performance. We need the devices to work at high current density so that we can make the chip size small and very low in cost, which is beneficial to consumers. And then for all of us, we would like our appliances to last for a very long time, which means we want reliable operation, and that requires the switch to be very rugged. Next. So to make you understand how the switch performance has changed over the years, I'd like to trace the evolution of power semiconductor devices on this slide. And I put the switch requirements at the bottom of the slide to remind us of what we are trying to achieve. Next. So you can see here the early power devices were bipolar transistors introduced by the industry in 1950. These devices were used for a while, but they were not voltage control devices, and they had poor switching performance and not very good ruggedness. Next. So in the 1970s, the industry introduced the power MOSFET. This device is a voltage control device with very high switching speed and excellent ruggedness. And this is widely used even today, but it could not handle high power levels. So you find this device in places like your personal computer in the power supply and for lower power applications. So to use devices in higher power applications, such as appliances in your home or in electric cars or in locomotives, I decided to invent a new device. Next. This is a device that I named the IGBT, which was invented in 1980, and it combined the physics of the bipolar transistor with the MOSFET for the first time. Next. The device was very rapidly commercialized by me by taking it from concept to product in less than 10 months. And this is a very unusual occurrence because in industry, it normally takes many years, 3, 4, 5 years to bring a new idea into the marketplace. The patent for this was issued to me in 1990 after GE filed for the invention in 1980. But in the meantime, it was very rapidly adopted at GE for a large number of applications because GE was such a diverse company, and they built the first heat pump drives, first innovative lighting, appliance controls, medical devices, and so on using this device. Next. But to achieve this, it was not easy. And since you are mostly young engineers who are aspiring to get into a new field, I want to point out that creating a new idea comes with a lot of difficulty. So let me share some of the problems that I had to overcome in order to make this device successful. So the first problem I had to overcome is that inside this device, there is something called a parasitic thyristor shown in the red box. And skeptics believe that this parasitic thyristor would destroy the performance of the device, preventing it from being functional. So I had to add some new innovations, such as the deep P plus region, which is in one of my patents issued in 1984. And with that, I could move the latch-up current density to a very high level, well above the normal operating range, making this device functional. Next. After I achieved that, the skeptics felt that this device would have very limited use because it would switch too slowly for most applications. So if you are running appliances off a 50 hertz or 60 hertz source, the device can switch slowly, and that's acceptable. But if you want to make an adjustable speed drive or an electric vehicle drive, the device should be able to switch at 10,000 hertz or maybe 20,000 hertz. And to do that, it was necessary to do what we call lifetime control. And at the time I invented this device, there was no known process to do such lifetime control for a MOS control device. So I had to create a unique process which I discovered with a post-electron radiation anneal to solve the problems of the electron radiation damage. And without this, we would not have the kind of devices we have today. Next. The other thing I showed in the early days was that this device had excellent high temperature operation. And this was documented in numerous papers that I published. The importance of this is that in most applications, such as in your automobile, the environment is very hot under the hood of the car. So if you want to use this device for ignition control, it has to tolerate that high temperature ambient. Another place where this happens is in the base of a compact fluorescent lamp, which is shown in the middle of the slide on the right-hand side. In fact, GE even used this to control steam irons, where the device had to tolerate the heat coming from the base of the iron when you're ironing clothes. So this was a very important discovery in order to make this device highly application dominant and to be used in a large number of applications. Next slide. So this is shows you a historical milestone where GE announced a product in June 1983 with a data sheet shown on the left-hand side. At that time, in the early days, we called this device the IGT, insulated gate transistor, which was later renamed insulated gate bipolar transistor, or IGBT. And this device won a prestigious award from the Electronics Products magazine as Product of the Year. We also published an important paper in a conference to describe how this works so that the people in industry would begin to use this device for many applications. Just as a historical note, this is about 2 years before companies in Japan and Europe began to create IGBT products. Next slide. Another important thing we showed in the early days, in the early 1980s, is that the device structure could be used to scale up the voltage rating very rapidly. This is important because if you scale up the voltage rating, the device is able to be used for many, many more applications. So if you have a device that works only up to 1,000 volts, it may be okay for electric cars, but it cannot be used for electric locomotives or for power distribution systems. So being able to scale up the voltage to the 6,000 volt level, as shown in this chart, for the planar gate asymmetric device, and later with silicon carbide to 15 kV devices, is important for expanding its applications. Next. So because of this ability to scale the voltage and to also parallel chips to make power modules with higher current handling capability, we could get an exponential growth in the power handling capability as well. And that was very important because the device could then be used in bullet trains like the Shinkansen bullet train in Japan or any other high-speed train around the world. It is even used today for ship propulsion and power distribution systems. Next. So the device today is used in practically all sectors of the economy. Next. In the consumer sector, you use it in your home for flat cooktops, for all your small appliances, for your washing machines, for your microwave oven. It is used in the transportation sector to make electric cars, which I'll be talking about more in this presentation. It is used, as I said already, in high-speed bullet trains. In the medical sector, it is used in X-ray machines, CAT scanners, MRI machines. All the power supplies are based on IGBTs. And one of the interesting applications that developed was the portable defibrillator using IGBTs. And that is saving more than 100,000 lives every year just in the United States. It is used in the financial sector to make UPS systems for very reliable financial transactions. And then in the lighting sector, it was used to create the CFL lamp, which I'll be again discussing later in the presentation. In the industrial sector, it's used a lot for motor control and in particular for robotics. And then in this presentation, I want to talk to you about how it is used also in the renewable energy sector, which is very important if we want to mitigate carbon dioxide in the atmosphere. Next. So I borrowed this concept from a paper by Kintish. Next, he says that to get improved living conditions, we need to provide people with more goods and services, but this results in more energy consumption, which of course causes more carbon dioxide emissions. That increases carbon dioxide in the atmosphere, and that of course results in global warming and climate change, which of course degrades the ecosystem. So we are not able to improve our living conditions under this scenario. Next. We can break this problem by doing conservation, which means using less goods and services, but that's not very popular among people. So instead, what we need to do is deliver these goods and services with greater efficiency, and we need to use renewable energy sources in place of fossil fuels. Otherwise, we would have to do carbon sequestration to reduce the carbon dioxide emissions, or do geoengineering. And eventually, if none of this works, then we have to do adaptations, such as building high seawalls to prevent the ocean from entering into our cities. In this presentation, I want to talk to you about the impact of how we have improved efficiency to solve some of these issues. So next. So in terms of IGBTs, we have obtained great improvements in efficiency in 3 areas that I'll highlight in this presentation. First is efficient air conditioning and refrigeration. The second is efficient lighting. And the third is efficient transportation. Next. So here we'll begin by talking about transportation. You can see here various modes of transportation and the percentage of fuel that is consumed by these modes. Next. So you can see that about the internal combustion engine in automobiles for trucks and cars consumes about 80% of the fuel around the world. Unfortunately, burning 1 gallon of gasoline produces 19.4 pounds of carbon dioxide emitted into the atmosphere. So one of the important things we need to do is to reduce consumption of gasoline. Next. So to understand how we can do this, let's go back to how cars were originally built by using the Kettering ignition system. This is a mechanical system that has been used for many, many decades. It is the basis for driving the spark plug in your internal combustion engine cars. And that is done by using a distributor, which is a mechanical system where something rotates and makes contacts to the various spark plugs. But when the spark plug is ignited, it also causes an arc flash in the distributor, and that wears out the distributor, causing poor timing, and you have to do constant tune-ups. And even with that, the fuel efficiency is not very good. So the next slide shows how a great improvement was made possible by replacing that distributor by using electronic ignition systems with IGBTs. So here the IGBT is being used to control the current flow through the ignition coil and deliver it to the spark plug. But what we can do with the IGBT is electronically control both the current and the voltage, and this wave shaping allows much more efficient use of the fuel inside the internal combustion engine. Next. Today, many companies produce IGBTs just for this one application, and billions of IGBTs have been sold by numerous companies, including Fairchild that I've illustrated here. Next. So to understand the impact of this on society, let's take a look at how much gasoline we use. And I'll focus today on the worldwide numbers. So you can see at the top of the chart that in the world, we are using approximately 600 billion gallons of gasoline every year. And this has sort of flattened out in year 2015. So I can extend the data out to 2020 by using the numbers in 2015. Next. So if you take the information that the electronic ignition system improved fuel efficiency by at least 10%, you can then calculate how much gasoline savings was produced from this chart, and it works out to be an enormous number of 1.79 trillion gallons during the 30-year time frame from 1990 to 2020. Next. So as I said already, burning 1 gallon of gasoline produces 19.4 pounds of carbon dioxide. So if we don't consume the gasoline, as pointed out in the previous slide, then we also eliminate carbon dioxide emissions, and that is calculated on this slide. So it shows that every year we have been now reducing carbon emissions by about 1,200 billion pounds. And we can then add that up over the 30-year time frame. Next. From 1990 to 2020, and it amounts to a very large number of 34.7 trillion pounds of carbon dioxide not being emitted into the atmosphere because of the electronic ignition system using IGBTs. Next. Now turning our attention to air conditioning and refrigeration, I want to point out that motors are very prevalent in our society and consume about 2/3 of all the electricity that is delivered in the world. And this DOE report, next, shows that for the residential sector, we use this extensively in our refrigerators and heat pumps. Next, in our commercial sector, it is used for vending machines and unitary air conditioners, which are basically large air conditioners for buildings. Next. So the old technology by which this was done was to take the input AC power that you receive in your homes. In the United States, that's at a fixed frequency of 60 hertz. And in other parts of the world, it may be 50 hertz. But that is then fed into an induction motor, which is very efficient, 95% efficiency. But once you feed this into the induction motor, it rotates at a fixed frequency. And that cannot be fed into loads, such as fans or pumps, where you want to regulate the load, change the amount of air that's blowing or the amount of liquid that's being pumped. So in order to regulate that, people were using dampers, but dampers convert the energy that's not used into heat. So this is a very poor in terms of efficiency, and the efficiencies obtained were less than 50%. Next. So the new technology that we developed at GE was the adjustable speed drive technology based on IGBTs. Here, as you can see in the lower left, we take the AC input power and then we use the adjustable speed drive containing IGBT switches to produce power at a variable frequency. So instead of having the fixed 60 hertz frequency coming in to the home, we are now able to convert that to a variable frequency, which can be anywhere from 60 hertz upwards to even 20,000 hertz. And we feed that into the induction motor, and it then spins in proportion to that new frequency. And that then allows us to apply it to the load and obtain various functions at the load level. So since we eliminate the use of dampers, the efficiency of this system improved by more than 40%. Next. So to illustrate what we did, I found some slides from 1983 in my archives. It shows the first IGBT built for ASDs using my design in 1983 at the top, and the first commercial adjustable speed drives with a motor at the bottom. Next. So to understand the impact of this, we have to look at how much electricity is used in the world. And in 2014, it was 25,000 terawatt hours. In 2020, it grew to about 26,000 terawatt hours. But from 1990 to 2015, there was a steady growth in the amount of electricity consumed. So if we assume that 2/3 of this electricity is used to run motors, and we get a 40% power savings by using adjustable speed drives, then you can calculate the power savings. Now I've shown three scenarios here, one with 25% adaptation or penetration of ASDs in the market, another with 50%, and the last with 100%. Now adjustable speed drives cost more than the old technology. So a lot of people are not willing to pay for this technology because of the initial higher cost. So today, percent of motor drives are built using adjustable speed drives, but with the right uh, incentives, it is possible to shift that closer to 100%. Next. So if you assume these conditions, then we can calculate the cumulative worldwide energy savings for the case of 50% penetration from the year 1990 to 2020, and it turns out to be a very large number of 73,000 terawatt hours. Next. To understand the impact of this on carbon emissions, we have to appreciate that most of our electricity is produced using fossil fuels, as shown on the right-hand side. In that chart, it shows that using coal and natural gas, 68% of the electricity is produced around the world. Now, we also know that when we produce this electricity with fossil fuels, we produce 1.1 pounds of carbon dioxide for every kilowatt hour. So using this data, we can convert the energy use into carbon dioxide emissions. So that is calculated and shown on this slide. And what I've shown here is the amount that is reduced by the use of adjustable speed drives because of the improved efficiency by 40%. Next. So if you take this chart and calculate the cumulative worldwide carbon dioxide reduction from 1990 to 2020, it turns out to be about 80 trillion pounds if you take the 50% penetration scenario. Next. Now turning to the case of lighting, for a very long time, we relied on the incandescent lamp, which is a relic of the Edison era. In this technology, we heat a filament to about 3,000 degrees centigrade, at which point it emits light. But less than 5% of the energy is actually converted into light. The rest of it is converted into heat. As we all know, the bulb gets extremely hot. In addition, this technology has a fairly short lifespan, but consumers like this approach because it's inexpensive and considered disposable. Next. However, because of the inefficiency, lighting consumes about 1/5 of the electricity used in the world today. Next. To improve upon this, we had to make a shift to the compact fluorescent lamp. This was initially done by work done at GE in the 1980s using IGBTs to create new types of lamps, which they named Triad and Halok, which eventually became the compact fluorescent lamp, or CFL. The challenge making a CFL is that the electronics has to be very compact and must fit in the base below the tube of the compact fluorescent bulb, and then you can make a bulb that can screw into your regular lamp sockets. And because the electronics is close to the tube, there is a lot of heat generated, and so it must be able to tolerate heat as well. So you need very compact power electronics which can tolerate heat, which, as I explained before, is something made possible by using IGBTs. So using this approach, we are now able to build compact fluorescent lamps, which are 75% more energy efficient. In other words, a 60-watt equivalent lamp only consumes about 13 to 15 watts of electricity with a CFL. In addition, these CFLs have long lifespans, which I, of course, like and all consumers like because you don't have to change your bulb often. Next. To see the impact of this, let's look at how many bulbs are in use. The technology grew in volume in the 1990s rather slowly, but a lot of countries put in incentives for adaptation of this technology. For example, in Japan by 1996, 80% of households were using CFLs. In India, they put in a project to install 400 million of them. In China, 800 million CFLs were sold even way back in 2007. As a consequence, the number of CFLs in use has grown rapidly after year 2005. And today, next, we have worldwide 22 billion CFLs in use. So let's look at how this has improved carbon dioxide emissions. Next. So to understand that, we can convert CFL usage for 6 hours per day and energy savings of 45 watts for a 60-watt bulb, and then calculate the power savings. And you can see that the power savings by 2015 has risen to 1,000 gigawatts. Next. And what this means is that the energy savings have grown from 1990 to 2020 to 59,000 terawatt hours. Next. As pointed out before, for the case of adjustable speed drives, most of the electricity is produced using fossil fuels, which emit 1.1 pounds per kilowatt hour. So we convert that for the case of CFLs and find that the global carbon dioxide reduction has reached about 2.5 trillion pounds by 2015. Next. So if you add all this up over the time frame from 1990 to 2020, the cumulative carbon dioxide reduction comes out to be about 66 trillion pounds. Next. So here's a summary of the impact of this technology from 1990 to 2020. And I've listed the numbers for the gasoline and energy savings achieved. What I did not go through is a calculation of the cost savings, which are documented in some of my publications. But by using the cost of gasoline and the cost of electricity, you can calculate the numbers on the right-hand side column. And you can see that not only have we reduced energy and gasoline consumption, but consumers have actually saved a huge amount of money, $33.6 trillion, because they do not have to use as much gasoline and electricity. Next slide. So the worldwide utilities also did not have to construct coal-fired power plants, and they cost about $3 billion for a 1 gigawatt power plant. So by not having to construct 1,366 power plants, they have saved $4 trillion. Next. But probably most importantly, worldwide consumers have saved $33.6 trillion by not consuming 1.79 trillion gallons of gasoline and 133,000 terawatt hours of electricity. Next. In terms of carbon dioxide emissions, I already talked about the numbers on the right-hand side. And if you add those up, it comes out to be 181 trillion pounds over the time frame of 1990 to 2020. Let's put this in context. So next. So let's go to the next. So total global carbon dioxide emissions per year in the world amounts to 8,000 pounds per person multiplied by 7 billion people in the world. The 8,000 is the carbon footprint for an average person in the world, and that amounts to 56 trillion pounds. What this means is that global activity by human beings is producing 56 trillion pounds of carbon dioxide each year. Next. So what the IGBT has done with the electronic ignition system, adjustable speed drives, and compact fluorescent lamps is to offset carbon dioxide emissions due to human activity over a 3-year time span. And this is, of course, over a 30-year duration. Next. Now I want to talk to you about future opportunities to improve things for mankind. The first is to deploy renewable energy sources. And this is, of course, being done by 2 methods. The first is illustrated here, which is solar. As you can see from the chart, solar deployment has been growing very rapidly, which is very good to see. Next. So this is done either in the form of solar farms, which are large power generating stations, or even rooftop solar panels in homes and businesses. Next. All of these are done by converting the DC power produced by the solar panels into the AC power required to be fed into the grid by using IGBTs as shown in the inverter circuit. So what the IGBTs do is to convert the solar power, which is DC, into the well-regulated 60 hertz power that can be used by consumers and industry. Next. Let's take now a look at wind power. Wind power has also grown rapidly in the last few years, particularly from year 2000, as shown by the chart on the left-hand side. Next. In the case of wind power, the source of energy comes from the wind turbine. As it rotates due to the flow of wind, it produces electricity through a generator. But that is a variable frequency AC power that varies during the day, in the night, depending upon how hard the wind blows. To be able to use this electricity, we have to convert it again into a very well regulated 60 hertz AC power, and that's done by using IGBTs as shown in the left-hand side. The advantage of using the IGBT-based inverter was it eliminated the gear train, and that greatly reduced the weight and size, allowing all the electronics to be located at the top of the wind tower nacelle. Next. There's an interesting quote here from Electronic Design magazine going all the way back to 2004, which said that the progress within IGBT technology has made the biggest single contribution to wind turbine advances over the past several decades. So having these IGBTs has been crucial to the success of wind power. Next. Now I want to turn our attention to other opportunities, in particular to transportation. And that is in the case of hybrid electric and electric cars, which all use IGBTs for the designs. So one of the earliest examples of hybrid electric car was the Toyota Prius. In this car, the IGBT is used, as shown in blue, for the electronic ignition system of the internal combustion engine. And then it is also used in the inverter in red for delivering power from the battery. So you can see in this chart, can be used in the electric only mode or the hybrid electric mode, or it can be used to charge the battery, and it can be even used for regenerative braking, which is very important for getting better fuel efficiency. Next. Today, almost all manufacturers have committed to making electric cars. In fact, many of them have committed to completely going from internal combustion engines to all electric cars by 2035. To make this successful, however, it is important that we deploy a large number of charging stations, but these charging stations should not be powered by fossil fuels. They need to be powered by renewable energy sources. Next. So if we do this, then you can see that we've been consuming 600 billion gallons of gasoline, and we can eliminate all of that. So next. And if we are successful in doing that, then we can reduce annual carbon dioxide emissions by 11.6 trillion pounds, which would be tremendously advantageous. Next. In the case of adjustable speed drives, we can provide incentives to move from the 50% penetration to 100% penetration. And this will again greatly improve carbon dioxide emissions. Next. So next, yes. So you can see here that by shifting to 100%, we can reduce carbon emissions by 3.5 trillion pounds each year. Next. And finally, if you take a look at electricity use, as I said before, we are using about 25,000 terawatt hours of electricity. Next. If we take all of this electricity that is produced, 68% of that comes from fossil fuels, which is 17,000 terawatt hours. So if you take all of that and we convert it into renewable energy, then we could save carbon dioxide emissions of 19 trillion pounds. Next. So to come to the end of my presentation, I'd like to give you some takeaways. First, power devices, in particular IGBTs, have already made a very large impact on efficient utilization of energy in all sectors of the economy. Carbon dioxide reduction achieved over a period from 1990 to 2020 is 181 trillion pounds. The IGBTs also play an essential role in all renewable energy, power generation, and are critical components for hybrid electric and electric cars. So we have a future where we could reduce carbon dioxide emissions tremendously by going to 100% electric vehicles, 100% adjustable speed drives, and 100% renewable energy generation. And this could reduce annual carbon dioxide emissions by 34 trillion pounds. So my message is that we have technology and we have the know-how to make big impact on reducing carbon dioxide emissions to mitigate global warming. It is a matter of proper investment and proper political strategy around the world to make a commitment to making this happen. So I hope you found this story inspiring, because it shows how a single technology developed a long time ago in 1980.
Time ago.
Invention has had very positive impacts on many facets of our modern life. So now it is time for question for our young scientists out there. And there's one question that I really like, because this is my question actually. And I think in your talk, Professor Baliga, you mentioned about skeptics twice at least. So the question really is that how do you deal with skeptics? Okay.
So can you hear me? Are you able to hear me?
Yes, we can hear you.
Oh, okay. Okay. So yes, of course, you have to overcome skeptics by the strength of your ideas, and you have to be a very good salesman, not only a scientist, but someone who can make a compelling argument to your skeptics that it is possible to overcome their objections. But it takes a lot of conviction, meaning you have to, first of all, believe in your idea very strongly and have done enough groundwork to have the facts straight so that you can overcome these objections. But I can mention something interesting. I like to quote Arthur C. Clarke, who is a famous writer who wrote 2001 Space Odyssey, which became a very famous movie. But he's also known for creating the geosynchronous satellites. That was his idea, and it was implemented, and we use it for communication today. But he said that when you propose any new idea, there will always be people saying it is impossible. And then once you show that it is possible, they'll say, oh, well, maybe it's not worth doing. We'll just keep doing what we are doing. And then finally, when you show that it really works well, they said, oh, yeah, that was my idea after all. That's why it works so well. So be prepared for that kind of response. But you know you have been fully successful when other people start taking credit for the work you have done.
So I hope this is useful for Elwin Jia. So the next question actually is a combined question from two audience. It's actually from Hafiza and Sri Kunal. And the question really is that you have covered a lot of application about IGBTs in our modern life. I can mention a few: consumer electronics, energy, lighting, transportations. Would you be able to comment on the application of IGBT devices for medical as well as the IoT sectors?
Okay, so in the case of medical, one of the places which I was very surprised to see the IGBT used was to create the portable defibrillator. So if you're not familiar with this, this is a small laptop size unit that people have now created. And inside that, it is a very highly automated system so that if someone is suffering cardiac arrest, you can quickly get this defibrillator from a box in your building, or it is now deployed in all the airplanes. So even when you're flying, you can quickly grab this box and you can then respond to the person having cardiac arrest. In the case of victims of cardiac arrest, you must respond within 10 minutes to save a person's life. So it's very important that this be in close proximity. So that is only possible if these were highly portable, of course, and it became possible only by using IGBTs, because when you do the shock, it is a very high voltage, 2000 volt biphasic shock that's given to the cardiac arrest victim, and only the IGBT could handle it. So that was a surprising innovation for me, because I developed it to run motors or lighting and so on. Let me mention that it is everywhere in the medical systems. When you go to the hospital, like X-ray machines in the power supplies, it is used in MRI machines, in CAT scanners, So it is very much prevalent in medical systems. If you get a ultrasound, perhaps you've had a baby and you wanted to see the image of the baby before the baby was born. That's ultrasound system where the IGBT is used to control the transducer. So yes, it is very prevalent. I should mention that I've written a book on the IGBT, which came out in 2015. And if you're interested in details of all of this, it's explained in a 700-page book. So please get that book if you're interested.
Okay, fair enough. The next question is from JJ. Okay. Now his question is that is there any more room left, okay, for advancement and performance improvement in IGBT devices?
So the IGBT was a huge quantum leap in performance, as you just heard, and it was revolutionary because it combined two different kinds of physics, MOS physics and bipolar physics. And we made this out of the traditional silicon material. But curiously, I invented this in 1980, as you heard from my presentation. But that same year, it was a very good year for me. I also created something known as the Baliga's figure of merit. This is an equation quoted by everybody now. It was an equation I derived based on theoretical principles that showed that if you change from silicon material to wide bandgap semiconductors, in particular silicon carbide and gallium nitride, then we could improve performance by not just 10 times, which is huge, but by 1,000 times. Okay, so it was an enormous prediction that we could greatly improve performance. So yes, there is a way to improve performance. However, the material was not ready. The quality of the material was poor. The size of the wafers was small. So we could not go from the concept, the idea, to products very quickly. So it has taken about 30 to 40 years of work to bring the material up to the quality we need. And that has finally happened in about 2010. And silicon carbide products have started appearing in 2010. And the big challenge for us since that time is to bring down the cost of the silicon carbides, because it is not enough to have performance. We need also reasonable cost to have a solution for the industry. So I've been working on this at the Power America Institute, which was founded through funding by President Obama back in 2015. And in that institute, I've created the national United States process for making silicon carbide devices at a foundry, and we are successfully making devices there. A number of companies have also created their own processes and are producing devices there. So we expect this technology to become more and more prevalent, and it won't completely replace the IGBT because it's such a good silicon device and its cost is very low. But for some applications where performance is also important, higher performance is needed, then these silicon carbide devices will indeed start getting used.
Okay, very good. So this is the next question from Yamuna. I think it's basically it's a dilemma that most I think young researchers are in. So the question really is about, as a young researcher, should you be focusing more on uh, new concepts, or should you be focusing more on product development? Any
So
advice?
yes, so it's always better to get into a new area where new concepts can be developed because it's very fertile ground. You can do that original work and publish papers and hopefully develop a good reputation. But let me just say that in my own case, when I joined GE, it is not, I did not want to work on power devices because they were already 25 to 30 years old. And I thought everybody had already done things in power devices that could be conceived of. But that was the only job I was able to get, so I was forced to work in that area. And fortunately, I was able to create all these new ideas like the IGBT and many others, which we haven't talked about because I have 100 plus patents. So, but those ideas helped transform the power electronics field, transform the power semiconductor field. So it was still possible, even though it was fairly mature, to make another big quantum leap in performance. And we're still doing that with the wide bandgap materials. So I wouldn't discourage you completely from going into a mature field. If you're brilliant and have ideas, you can still make a contribution. But it's easier if you can go into a new area. Let me answer the other part of your question, which is that when you're doing these innovations, my personal view is that you should keep an eye on product and impact on consumers and humanity. So the best work you can do is if whatever you innovate actually comes and gets used by people in the world and benefits people. So for me, the greatest satisfaction has been that a device like the IGBT has had so much impact on people as we went through in my presentation. So hopefully each of you can come up with these novelties and improve things for humanity.
Yeah, I hope that's useful. So maybe we'll go to the last question about, future density scaling for IGBT devices. So Chua Wu basically proposed an idea of going 3D stacking, but he also worried about heat dissipation. Do you have any comments on this idea?
So the way we do it with the IGBT is because it has low on-state voltage drop and high current density, we are able to make small chips, and you have to be able to remove that heat. And there's now very mature packaging technology to allow us to do that. And we can parallel lots of these chips to get to high current levels. But that's now become fairly mature. So if you go to silicon carbide, we need to rethink that and improve the packaging just for silicon carbide as well. And there we are also moving up in frequency. So it's not just heat removal. We have to worry about electrical parameters like stray inductance. Otherwise, you get all kinds of problems. So yes, packaging, etc., is very important in our area. And we continue to make great enhancements in that area as well.
Okay, I guess that is all the time we have for Q&A. But I hope this talk have inspired all the young scientists out there. Okay, so that you can you can aspire yourself to be the next leaders in research. So on behalf of GYSS, let me thank Professor Baliga for your very insightful sharing. Thank you very much and have a good day.
Dear guests, it is a privilege to have with us today Professor Leslie Valiant, and he will present a plenary topic entitled Where Neuroscience Meets Computer Science. Professor Valiant is currently the T. Jefferson Coolidge Professor of Computer Science and Applied Mathematics at the School of Engineering and Applied Sciences at Harvard University. His research has spanned multiple areas, ranging from theoretical computer science to parallel computation, as well as artificial intelligence and computational neuroscience, among others. He's also an author of 2 books, Circuits of the Mind and Probably Approximately Correct. He's a recipient of the Neven-Lena Prize of the International Congress of Mathematicians, as well as the 2010 AM Turing Award. He's a fellow of the Royal Society and a member of the U.S. National Academy of Sciences. Let's welcome Professor Valiant.
Thank you very much for the very kind introduction. And thank you to the organizers for inviting me. And thank you, the audience, very much for coming. Science comes in many varieties. So some projects are short, you can complete it in a month. Some take a year, some take much longer. So the one I'm going to describe, I've been doing for more than 30 years. And it's not quite finished. So it's very much an ongoing project. Some science projects are in one specialized area, and most of them are, and there are good reasons for that. And some projects try to bridge different areas. So the one I'm describing obviously tries to bridge 2 areas. And so in my experience, this is quite difficult because although people describe how specialists in different areas can come together and do productive work, in my experience, it's usually the single scientist who has to try to understand more than one area, and that can meet with difficulties. So what this talk is about how computer science may be able to help us understand how the brain works, So neuroscience has a very rich set of experimental techniques for finding out about the biology of our brain. But to describe at a high level what functions the brain is doing, the right language for that is computer science. So this is an ideal opportunity for these two fields to interact. So the point about the brain is that while a vast number of things are known about it, it's amazing how little we know. So a very basic question of how many neurons we need or we use to store a memory of, say, one event, or maybe one person. So if at GYSS you meet a new person and you remember the person for a while, how many neurons do you devote to this task? Is it 1? Is it 10? Is it 100? Well, we have about 10 to the 11 neurons, so maybe it's as large as 10 to the 8. So it's unlikely that the answer is 1 or 10, but otherwise we really, just do not know. And this is astonishing level of ignorance on such a very basic question. And the question is, why is this so difficult? And so the approach I'll take from computer science, the methodology is the following, is that if you take an easy problem, like storing some information for a computer is very easy. And if you name such a problem, then maybe there are so many different ways of implementing it that it's hard to discover which one the brain has chosen. And that seems to be the case, that we haven't been able to discover how the brain does this. So this methodology is to invert this. And the idea is that if there are fewer ways of implementing something, if it's something hard to compute, then perhaps this gives us a clue. It narrows down the possible solutions the brain might be using to implement this. Okay, so this is the idea. And just to illustrate this, so this is an illustration of some interaction between a cognitive task and computational difficulty. So this task is, for the light bulb problem, it's an, let's say, imaginary task. So there's a large number of light bulbs, maybe 1,000 in front of you, and they're each flashing on and off with a period of a second. So if you look at one light bulb, it's either on, it may be on again next second, then off. So each one is randomly on and off with a period of 1 second. And the setup is that each of these 1,000 light bulbs flashes independently, except 2 of them are correlated. So 2 of them will be the same 3/4 of the time, say, and not half the time. Okay. Now, of course, if you just stare at one light bulb, you will just see the sequence of zeros and ones, whether it's off or on. And if you stare at another light bulb, you see that's zeros and ones. And so if you compare these two particular sequences, then after a while, you'll figure out whether do they agree about half the time, or do they agree about 3/4 of the time? Okay. But the question is, how difficult is it to tell which pair is correlated? So could you do this for a large number of light bulbs? And so just to put this down, so you've got a large number of light bulbs, each randomly flashing off and on, and they're all independently flashing, except for one pair, which you don't know which pair, and that's correlated. And your job is to find the pair. And the obvious algorithm for doing this would be that you have to focus on each pair. You focus on a pair, you stare at it for a while, look at the zeros and ones, and see whether they agree half the time or 3/4 of the time on the average. But then you've got about n² pairs of light bulbs to stare at, and that takes a long time. Because if you think about it for not very long, in fact, the time you have to observe one pair of light bulbs to see whether they're correlated is just the logarithm of the number of light bulbs. And if you have about some constant times the logarithm of number of light bulbs, zeros and ones, and you observe each light bulb for that long, then that will have enough information in to determine which pair is correlated. So what I'm saying is that for each light bulb, you write down a sequence of zeros and ones of about log n bits, and then you go away and do your computation, compare each pair. But the point is that the obvious method is n squared. In principle, the amount of information you have is much smaller. So the question is, is there a fast way of doing this? Faster than n squared? And the answer is that people have thought about this for a long time. There are various algorithms, and the best algorithms are better than n squared. You can get them like n to the 1.6, but they're very complicated. Okay, and certainly there's no easy way of doing this, as far as we know. So this looks like a problem with some inherent computational difficulty to the best of our knowledge. So the question is, what does this mean as far as cognition? Well, I think what it means is that I'd be amazed if a human could do this problem on a large scale. Because, of course, it's hard to exclude. It's possible that the brain is especially wired to do this problem, but it's unlikely. But so it looks like, without carrying on about this anymore, so I think this is a problem with some inherent computational difficulty. And for that reason, we are bad at it. Okay, so given 1,000 light bulbs, I don't think anyone could solve this problem very well, except by staring at each pair of light bulbs in turn, and that'll take a long time. Okay, so this is a problem which I think is too difficult, we cannot do. So this is not interesting. So what problems are interesting to help us find out how the brain works? And so I'll look at symbolic processing. What symbolic processing is that we can tell each other lots of stories about discrete facts, and the other person can just soak it up. Okay, so if I say that there's a restaurant called the Green Fox, and I'll tell you that I went there, it was bad choice. It was recommended by Joe. When I was there, I heard the election results. Then you can soak this up, you can remember some of this, and there isn't any great mystery. But the point is that humans can do this kind of thing on a very large scale. We can do this every day, and we can remember much of what we did the previous day. And there isn't any very good explanation or theory of what happens in our brain to do this. The point being that just one single act of memorizing something is a too easy a problem. But if we observe that the brain does much more, it can do this at scale, then it's a more challenging problem, and we have a better chance of narrowing down how the brain may do it. And here already we've got one feature, which is kind of a standard feature in psychology. The idea is that we don't just memorize things as a long list, but we structure it. So chunking is the idea that sometimes from two concepts like green and fox, we chunk it into a single concept, the Green Fox restaurant. And if you go to a restaurant often enough, then it becomes a primitive concept for you, just like fox was before. That somehow in our brain, not just like having a telephone directory, there's more structure there because some high-level concepts we need to be able to access fast, just as we access low-level concepts. Okay, so human cortex can do these kind of tasks hundreds of thousands or more in succession and remember things. And there's no viable theory known, even in principle, of how the brain actually does this. Okay. So now we can actually break down even more on why what the brain does is impressive as far as tasks, and have an even more onerous list of things which we have to explain. So I have a list of 7 here. So one-shot learning. The idea is that after one exposure, you can memorize things. You don't need thousands of exposures. We can do lifelong learning, that we can execute many of these tasks, maybe 100,000, maybe more. So how many things we can memorize in a lifetime? It's a good thing to try to estimate. Also, when we memorize something, it's not just that we it's like recording it somehow on a computer, we're also able usually to respond to it very fast. So we can recognize what we've learned years later and very fast. So what seems to happen is that there's some little subcircuit in your brain which incorporates this new information and can execute very fast. So the memorization is a particularly active kind of thing. And furthermore, these subcircuits, these various things which you've memorized, you can even compose together on the fly. So given a situation, you can compose two pieces of information you've learned in the past on the fly. So these subcircuits you've learned must have been learned very well. So you can just run with the information. Okay. Now, if you want a theory of all this, you want it to be quantitatively plausible. So we only have so many neurons. They only have so many connections. And the strength of connection is very important, as I'll mention, how much influence a neuron has on another neuron. Also, there's some basic facts about the brain which you have to be faithful to. And one thing is that the long distance connections in the brain, the axons, are one-way connections. So you have to be faithful to these things if you want the realistic explanation. And then, so the last thing, which is maybe become clearer later on, is that what I really want to do is to break up these tasks, which I haven't described very accurately yet. I've just given examples of this restaurant. And I want some small set of primitive tasks and explain how the brain does those and go from there. Okay, so so in this talk, on the one hand, I'll describe what I want this model of the brain to do. And on the other hand, I'll describe some constraints we know about the brain, the difficulties. And these two sides somehow have to come together and match. So there are some very obvious things to say about the brain. The computers have addressing mechanisms. There's a location 137. Brain, as far as we know, there's nothing like that. So the brain is very slow. Maybe 10 milliseconds is the cycle time. So in 100 steps, although it's got an enormous amount of parallel processing, in 100 steps, you can do amazing things. So the algorithms you need are very different. So each neuron is connected to a very small fraction of the other neurons, so it's sparsely connected. So one simplifying aspect of the brain, not everything is complicated. One simplifying aspect is that long distance communication is very stylized. There's just one way of doing it, and these are these spikes. So basically, as far as long distance communication, information is just carried by the time a message arrives. There's nothing in the message except for the time at which it arrives. Okay, and then very important are these quantitative constraints. You've got a fixed number of neurons, a fixed number of connections from each neuron, and then this is important that you also have to take into account how well neurons can talk to each other. So synaptic strength. So synaptic strength of 20, K is 20, means that each synapse is only a 20th of the strength needed to cause the neuron to fire and communicate. So if K is 20, it means you need at least 20 neurons to make one neuron pass on this message. So if K is small, then that's a stronger computer, but the brain pays a high price for having that. Okay, so as far as what's known about the brain which is relevant, so these are some well-known results from about 20 years ago, which show that there's some things which are known. And so this is what are known as Jennifer Aniston cells or the Bill Clinton cells. So the idea here is that you've got a patient, a human patient, who's got electrodes in certain parts of their brain, usually for medical reasons, but then the cognitive scientist comes in to do some more experiments. So this patient is looking at a picture on a computer, say this picture, And it's looking at this picture between these two dotted lines. And from one cell, there's a recording. This histogram shows you the strength of firing of a single neuron in the brain of this patient. And once you show this, so time goes on from left to right. Once you show this picture, then this cell fires. And then at this line, the picture is withdrawn. And then after a while, this cell stops firing, okay? And so what's found is that for this particular cell fired when it saw a picture of Bill Clinton, okay, even a caricature of Bill Clinton, but not when it saw a picture of a different American president or an animal or the Eiffel Tower or some buildings. Okay, so so how these experiments are done is that earlier, someone shows this patient various pictures and records from some neurons, and then starts suspecting that one neuron responds to Bill Clinton, and then in a separate session collects together pictures, some of which are of the suspected concept and some related concepts, and can somehow verify that in fact this cell, as far as appearances at least, this cell seemed to respond pretty well to Bill Clinton and not to other concepts which you may think are related. So the relevance of this, so this only works in certain parts of the brain, but the relevance is that if you expect to find a cell to respond to some concept like Bill Clinton, then this will only happen if you've got a very large number of cells which do correspond to Bill Clinton. Because if only 1 in a million cells correspond to Bill Clinton, then you'll never find it. So this suggests that large number of cells, the large numbers of cells which correspond to concepts and to natural concepts, which we think are human concepts like a person, we can find cells which correspond to them. Okay, so so that's okay. So what's the approach we take if there are no viable theories known to a problem like this? So we have to make up a model neural model which respects what we believe happens, and it'll underestimate the brain. So the fact that it's simple is okay as long as no one complains that our model is too powerful. So then we think up some challenging tasks, and then we show that these tasks can be executed on our model. And then we show furthermore that, in fact, our model can execute thousands of instances of these tasks. Okay, so these will be some called learning and memorization tasks. And so basically we need a model which no one will complain overestimates what the brain can do. As long as we can demonstrate in theory as an algorithm that this model can do it, we'll have one explanation of how it might be doable. Okay, so I'm still trying to explain these kind of things. And I'll explain this is ongoing work still. And the question is, what are these tasks which I want to do, which represent these more particular examples I've given? So I'll just give two examples. And one is allocating storage for a new concept. So at one time, for the first time, you heard of the notion of an Omicron variant. Okay, so you were blissfully ignorant up to some moment when you first heard of this. So what happened in your brain? Or more generally, what's the task? So the way I formalize the task is that to say that these two words, let's assume you've heard these two words before, Omicron and variant. If not, then there was another stage of learning. But supposing these two words you learned, and the task of memorizing this concept was to memorize a single concept made out of these two. It's like a chunking. Okay, and in particular, so the definition is that if you've already stored items A and B, which is Omicron and variant, then the task is to allocate neurons, to find some new neurons, to the item C, and change the circuits so that in the future, when both Omicron and variant were active, then you were reminded of C, because this new set of neurons were caused to fire. So this is the chunking. You're making an equal citizen of this new concept with your previous primitive concepts. Okay, it's chunking. So that's one task. And the second task is association. And this association is that you suppose now that you already know what the Omicron variant is, and you know what the word dangerous is, but you have been told that this variant is dangerous. So what happens in your brain? So the formalization is that you've got two concepts, A and B. A is Omicron variant now, B is dangerous, and somehow through your training, you'll, whenever you hear Omicron variant, you'll be reminded that it's dangerous. Okay, so these are two tasks, and I have two more, but okay. So now the model of computation I try to execute it on, I'll just summarize very quickly. So it's okay. So the main points are that I worry very much about resources. There's so many neurons, so many connections, and these are the strength of the synapses. So you have to keep within realistic limits of what the brain has. So the computation is split up into local updates. So each neuron has its own little algorithm which runs on it, and it's discrete. And what this neuron does depends on just what signals it gets from its neighboring neurons. But there's a description, so the local little distributed algorithm running on it. You have to explain all phenomena in those terms. And you also have to discuss timing, which is important in any computation like this. And basically, the timing assumption is very weak. It basically says that each neuron has like an internal clock, which approximately can is the same as that of neighboring neurons. And for a few steps, it can keep track of where the other ones are. It's a very weak timing mechanism. Okay, so just general comments. So it's known now that there are many kinds of neurons and even synapses. And we interpret this to mean that these different neurons are there for a good reason. They run different little algorithms. So we're not looking for the kind of electronic component model where you go out to buy just one kind of component and have clever ways of composing them to make different functions. Evolution has given us many different neurons to do different things which are useful because that's the most efficient way. And then the other point is that we also know that the brain is very communication challenged, that there are relatively few connections to what could be, and in some sense, all the neurons are waiting for information. And even if the neurons could do some vast amount of computation at each neuron, it wouldn't help that much. So it's communication challenged rather than computation challenged. So what happens at each neuron, defining that isn't so critical, getting that right, because the communication challenge is a limiting one. Okay, so the various issues here, but let's omit those. So I've told you most of the things you need to set up the problem. So for the association problem, what happens? So you've got one concept, the Omicron variant, another concept, dangerous, and what you need to do is to set up connections so that whenever this is active, this will be active. So here A and C are fixed. From what I've told you, I mean, there are these numbers, number of neurons, number of connections, how many neurons are devoted to concept, and how strong synapse is. And this defines for you the problem. So the problem is that when all of A fires, you want at least K of the connections to each of these neurons to fire. Now in this model, we assume the graph is like a random graph. And then this becomes a concrete mathematical problem, and you can analyze it, and you can see whether the graph can support this association task. Okay, so somehow I've given you all the information you need. The rest is just working out, and you use basic probability theory. So beta is this Bernoulli distribution. Okay, so you do that. So if you're familiar with neuroscience, there's much discussion in neuroscience about Hebb's rule. So Hebb's rule, so Hebb in the 1940s discussed the fact that if you've got two neurons connected, under what conditions does one, what happens if one excites the other? So in some sense, this is a generalization where instead of single neurons, you have sets of neurons. And we believe that at the systems level, this is how the brain works. So the other operation I mentioned are similar. Okay, so one can do computer simulations to figure out whether one can realize many of these tasks on a network. And basically, what we found was that there were two regimes. So in one regime, you've got fairly weak synapses. You need 16 nodes to fire to cause a single neuron to fire. But then the number of actions this brain could do was a few thousand. Okay, so the actions were sequences of these tasks, of associations, assignment of memories, etc., etc. And if you remember, we were aiming for hundreds of thousands. So it turns out in a different regime, if you've got very strong synapses, so this is almost like a computer. So k equals 1 means a single neuron can cause its neighbor to fire. Then you can go into territory you're interested in. For realistic numbers, you can get hundreds of thousands of actions supported. So this brain can memorize lots of things in sequence, and even after it's memorized 250,000, it's fairly reliable on the tasks it started with a long time ago. Okay. So some of these numbers, like the N's and the D's, etc., are known for simple neural systems, such as the locust, and one can make sense of those numbers. But what I want to finish on is the hope that one can actually test whether this kind of theory is right for the brain. So for the association task, so what's aggressive about the model is that the model assumes that if I have any two sets of neurons and run some protocol of exciting them, then I can have the effect of changing what the circuit will do. Okay, so the definition of this association task was that if I fire this set, then the result will be to cause this set to fire, to be reminded that the Omicron, if this means Omicron, variant, B means dangerous, by some training, whenever I think of Omicron variant, I will know it's dangerous. Okay, so the theory assumes that for random sets of neurons, after certain training, the brain is clever enough to figure out how to change the circuit. So there's a strong assumption. But fortunately, this is exactly the kind of assumption which in principle you can test experimentally, because you can poke electrodes or using optical techniques, probe random sets of neurons. So random sets of neurons are the easiest ones to probe. And if your assumption is very strong that this should work for random sets of neurons, then in principle, these are ideal for testing whether the brain supports this. And the experiment's ongoing, trying to figure this out. Okay, so so we're still trying to find explanations of why the brain does these very everyday things, which we all take for granted, but we can't explain. Okay, well, thank you.
Thank you, Professor Valiant, for a wonderful lecture. We have some questions from the audience, which I'll read off for you. The first one is asking, they've heard that deep learning can currently train around 100 hidden layers in a model within seconds. And they were interested in your thoughts on when you believe we'll soon be able to mimic the actual brain in cases of NLP or image recognition, for example.
Well, thank you for inviting me here for start. So I think the question presupposes that the brain works like deep learning, which I think is controversial and unresolved. So my bet is that the brain works rather differently from deep learning, although and in fact functionally is good at different things. So so one very basic issue people have raised is that clearly with backpropagation you need backward flow of information. You need two-way flow of information, which in the brain is hard to realize. Of course, people are trying to overcome that by having different kinds of algorithms. But so I would presume that deep learning is a very interesting engineering method, but it probably doesn't have that much to do with the brain.
Thank you. Next question. Can we make assumptions about non-neuronal components of the brain, such as glia and vasculature, using computer science?
Well, possibly in the long term. So what's attractive about looking at neurons first is that as I think I mentioned in the talk, is that the long distance communication is very stylized. So axons are all very similar and the transmission is all very similar. So there's one thing which is simple in the brain, is this long distance transmission of information by neurons. And this simplicity can be used as a constraint on the brain with which we can ask information. So I think everything else about the brain is just much more difficult. So I think those questions I haven't looked at, but I think we have to resolve what neurons are doing first, I think.
Thanks very much. The next question is why humans have a tendency to always remember bad memories or negative memories, incidents that happened a long time ago, but tend to forget happier moments easily. What are some of the underlying mechanisms? Your thoughts on that?
Okay, so that's not something I study, and I'm slightly surprised by the question. Is that true for everybody? I don't know. Are there studies of this? I'm not familiar with literature on this. I mean, people also talk about people suppressing negative memories. So I suspect it's a complicated phenomena, but I'm just not familiar with research in this area.
I'd imagine it's a very individualized process indeed. Yes. So next question. May we learn your opinion regarding emerging computational methods, such as physics-informed machine learning that is used to enhance drawbacks of conventional neural networks?
So repeat, what's the new technology?
Regarding, so your opinions regarding emerging computational methods, such as physics-informed machine learning to enhance the drawbacks of conventional neural networks.
So these are different. I'm not exactly sure what the question means, but physics-informed neural nets. So I'm not quite sure is the question about just different algorithms which you run on a conventional computer. So also I'm not quite sure whether is the goal to. I'm also not quite sure what drawbacks of neural nets one wants to overcome. So certainly trying to, if you have more information about the problem you're trying to solve. So neural nets are a very generic method of solving things. If you have more information, something called bias in machine learning, you may try to exploit that, but the successes in that direction are, I think, limited. So as far as I know, the powerful vanilla methods of machine learning, which are well programmed, are hard to compete with. But certainly there are millions of drawbacks, and everyone should try to overcome that. I'm not discouraging anything. I'm just always impressed by the most generic vanilla way of doing things in machine learning. Seems to do very well.
Thank you. So there are thoughts that no one ever really reaches their brain's full capacity. And can this be applied to cognition? So for example, if this is true, why do we seem to reach cognitive limits? An example would be multitasking.
Well, yes, I think well, they're two different things. So with multitasking, you're reaching limits of your working memory. So your working memory is a very special part of your brain, which is to do with your attention, what you're attending to, and that brings together information from all over your brain. It's a very complicated piece of machinery, presumably, so that's got strict limits. But I think the limits there are very different from what you use your kind of main cortex for. That, use your main cortex for remembering your experiences over decades and decades and decades. And ultimately, there must be limits to that. But so the idea that we're underutilizing our memory and we could always have memorized more, I mean, I tend to agree with. But certainly with the models I have, there is the tendency that it does tend to fill up the brain, but a lifetime seems to be short enough to not to fill it up completely, I think. So I think the answer to the question is that our short-term working memory has got very strict limits, and that's multitasking. But our long-term abilities to store information is a very different thing, and we have a very impressive capability for that.
Thank you. Another question. So when thinking about cognitive function, when you look at different patients or different people, as you mentioned, variability in the number of neurons, the number of connections, and synaptic strength. So it would seem that there is variability from person to person in how these factors drive their cognitive outcomes. And these processes alone may very well be dynamic over time for the same person. Can computer science play a key role in helping to understand the individualized nature of these parameters and maybe even optimize them over time?
Well, that's certainly the kind of effort I'm involved in. But relating it to reality is difficult because, I mean, these basic numbers, you would have thought that we understood what they were on the average for humans and what they were for chimpanzees and other mammals, but it's very hard to get hold of these numbers. So apparently experimental neuroscientists measuring these numbers doesn't bring them a great career prospect, so it's not a very fashionable thing to do. Whereas for people like me, these numbers are important. So it's a great question. I don't know the answer. So it could be that not differences in these numbers account for all kinds of mental capabilities or mental problems, but trying to understand the role of these numbers is exactly what I'm trying to do. But we haven't got the stage of saying anything about individuals yet.
To close, actually, a follow-up question. So when we think about computer science and neuroscience, two fields with certainly very different skill sets involved, and in our audience, we have our future innovators, our future scientific and engineering leaders in our community. What advice do you have for them in terms of in the future when they build their collaborations? Any advice on how to build strong collaborations, especially when they need other expertise to help answer the questions that they have? Any guidances on how to build stronger bridges in in multidisciplinary research?
Well, yeah, I mean, it's a fantastic question. So I've ventured in various directions to do this kind of thing. So one hears of this ideal story of people of different specialities sitting around the table and doing something together. But I found that to do anything, you have to understand the multiple fields in a single brain. that it's very hard to talk to people if there isn't some mutual understanding of the ground rules. So, for example, just one comment. So, in computer science, there's a long tradition of working top down, bottom up, appreciating many different levels of a system. Whereas neuroscience tends to be very bottom up science. That they look at they work up and they don't want to speculate on what they haven't demonstrated. So this is a very basic difference in outlook, which is difficult to bridge. So one will find these very basic differences in philosophy, which I haven't found a simple recipe for overcoming.
Thank you so much for sharing your wonderful lecture with us today and for answering our participants' questions. Thanks again for joining the
Ms. Rebecca Cooper is a graduate student researcher specializing in neuroimaging and mental health. She completed changes in sleep across the transition into adolescence, a period which is characterized by significant changes in sleep behavior. So the study also assessed the change and stability in sleep behavior in relation to emerging psychopathology symptoms during this key period in development. And ladies and gentlemen, please join me in welcoming Ms. Cooper.
Hello everyone, my name is Rebecca and I'm from Melbourne Neuropsychiatry Centre within the University of Melbourne. Today I'm going to be discussing sleep patterns in late childhood and adolescence and longitudinal relationships with psychopathology. Before I begin, I'd like to acknowledge the contributions of my co-authors as listed here. And in addition, I would also like to acknowledge the First Nations peoples where I'm presenting from today and the lands on which they are custodians of. So that is the Wurundjeri peoples of the Kulin Nations, and I acknowledge and pay my respects to the elders past and present. Now, when we focus on the transition from childhood into the early adolescence, we see that sleep disturbances are relatively prevalent. However, for those individuals that do have a relatively high level of sleep disturbances, these individuals also experience high levels of psychopathology. And this is just another term for poor mental health symptoms. Now, why this is really relevant is that individuals with a higher degree of psychopathology symptoms are much more likely to go on to develop a psychiatric illness in later adolescence or into adulthood. Now, if we look at the pattern or the specific sleep disturbances across this transition period, what we see is that the overall patterns of these sleep disturbances are in a state of flux. So we see a number of sleep disturbances become much less prevalent, and they're shown with the blue arrows there, while others such as insomnia become much more prevalent moving into adolescence. If we look at the psychopathology side of things, we see an overall increase in the experience of psychopathology symptoms, and we also see the onset of many psychiatric disorders during this period. I was really interested in the interplay between these two sorts of trajectories or changes, and I wondered whether they might be somehow interrelated. So the aim of our study was to assess or to investigate the changes in these sleep disturbance patterns further, and then to assess the temporal relationships with changes in psychopathology during this period. Now, data for our study was collected from the Adolescent Brain Cognitive Development Study, or ABCD study for short. This is a longitudinal study currently being run in the US in young adolescents. So we obtained data for just over 10,000 individuals at baseline, and this is when they were between 9 and 11 years of age. We also obtained data at the 2-year follow-up point. So here the individuals are between 11 and 13 years. We obtained a measure for sleep disturbance and psychopathology at each time point, as you can see detailed here. And for our main method of analysis, we utilized an approach known as a latent transition analysis. So for those who are familiar with k-means or clustering analyses, this model works in a very similar manner. But if you're unfamiliar, basically what this model will do is in a given population, it will identify whether there are subgroups of the population within this larger sample that have similar responses to a particular set of variables. So in our study, for example, we inputted the sleep disturbance data at both time points to determine whether there were patterns of different sleep disturbances that might cluster together within particular individuals. So for our results, we found firstly that there were 4 discrete patterns of sleep disturbances that were identified in both late childhood and early adolescence, and I'll just describe those briefly to you. So the first group that we identified were individuals that experienced an overall low level of sleep disturbance, and this at baseline, this group made up about 26% of the sample. The second group we identified had substantially higher levels, but sort of in the mixed to moderate range in terms of the severity of sleep disturbances that they experienced, and were thus the mixed disturbance. We identified a third group that had, similarly to the first group, a pattern of low levels of sleep disturbance across all symptom scales, with the exception that they had a very high severity of insomnia symptoms, and thus were the insomnia dominant group. And our fourth and final group were individuals that experienced high levels of sleep disturbance across all the different disturbance domains that we analysed. Now, what I find really interesting is that when we repeated this model at the 2-year follow-up, we found that the same overall patterns were experienced by those in early adolescence. However, what did change substantially was the proportion of individuals within each group. So what this graphic shows here is the proportion of individuals within each group that were reclassified into the same group over time, i.e. they didn't change in their sleep patterns over time. So as an example, those in the insomnia dominant group, we had 83% of those that were categorized in this group at baseline remained so in early adolescence. And this graphic also shows the proportion of individuals within each group that transition to a different profile. So for example, 6% of those initially in the insomnia dominant group transitioned to a pattern of high disturbance when they reached early adolescence. When we looked at the cross-sectional relationships with psychopathology, we found that membership in any of the more severe disturbance classes, so that's all of those other than the low disturbance class, were much more likely to develop or to experience symptoms of psychopathology. So what we generally found overall was that high levels of disturbance were associated with high levels of psychopathology, and this is shown in the plot here. So on the vertical axis here, we have all the different symptoms of psychopathology that we assessed in our study, and these odds ratios represent the probability of class membership in any of the 3 more severe sleep disturbance profiles relative to that low disturbance class. What I will point out is that regardless of which symptom we look at in relation to psychopathology, high levels of sleep disturbance were associated with all of these symptoms. So it seems to be a really like a transdiagnostic measure, or sleep disturbances are associated with a broad range of different psychopathology symptoms. When we move on to our longitudinal analysis, First, we wanted to ascertain whether psychopathology symptoms at baseline had any influence on the probability of individuals transitioning between sleep patterns over time. So what we're in essence asking is whether these symptoms of psychopathology in late childhood, whether they preceded these changes in sleep that we saw across our study. And we found using logistic regression that there was no relationship between these two variables. What we did find, however, was that transition between sleep patterns over time was significantly associated with changes in psychopathology, and what's more, they appeared to co-occur or co-localize over time. So what does that mean? What does that look like? Here I've shown this visually, but what we can see, or what to expand this a bit further, what I mean is that individuals that experienced an increase in the overall levels of sleep disturbance over time also experienced increases in psychopathology symptoms. And in reverse, those individuals that experienced a decrease in the sleep disturbances that they were experiencing also experienced a decrease in psychopathology. So to put in layman's terms, when one got worse, the other got worse, and when one got better, the other got better. And it seems to be in a very, we saw this effect mirrored across this timescale that we were assessing for. So just to kind of expand on that further within this graph, what I'm showing is that, for example, individuals in the mixed disturbance group that transitioned to high disturbance over time certainly had an increase in both internalizing and externalizing symptoms. And these are just different subtypes of psychopathology, I should add. While those, for example, that transitioned in the reverse direction, so for example, they began in the high disturbance class at late childhood, but transitioned to a pattern of mixed disturbance, we saw significant decreases in the psychopathology symptoms they experienced. So to wrap up and conclude, we found firstly that there were several discrete patterns of sleep disturbance present in both late childhood and adolescence, and these are in a state of flux over time. And secondly, that psychopathology symptoms do not precede these changes in sleep, but appear to co-localize with them. One interesting implication for these findings, and a field of or a venue for further research, which I'm really keen to look into, is to identify what predictors or what was different in these individuals that improved in sleep versus those that deteriorated in sleep. What kind of predicted those changes? What was the difference between improving or deteriorating? And thus, if we're able to identify some of these predictors, we could utilize these in
All right. Thank you very much, Jessica, for the very, very interesting talk. So we have some questions from the audience right now. So for the first question, yes. So did you consider any moderating or mediating factors for the transition across groups from baseline to 2 years later? For example, gender differences, whereby there is substantial evidence showing that there is significant gender differences between males and females across different psychopathological symptoms.
Thank you for your question. We looked broadly at whether the different classes had a different, whether the classes had different distributions in their sexes, and we found that the insomnia dominant group tended to have more females in it, and that was just at baseline and at follow up. But we didn't look at the moderating effects of changes over time, and we also didn't look at the relationships between the sleep scores and the psychopathology. But previous work that has been done in this data set shows that typically internalizing symptoms are more common in the females, and then in the males, they're more externalizing specific. So we didn't study it in our study, but I know from previous work that's what's been examined.
Okay, right. So another question that's come in is, The emerging patterns were very interesting. And have this been observed in adults or other cohorts of adolescents? And how do these patterns translate to a typically linear sleep screener?
I'm not sure what you mean by the linear sleep screener, but in terms of, this is the first study that's really looked at like latent patterns of sleep disturbances and how they change over time. They're not typically done. They're kind of an understudied kind of a underutilized kind of resource, I think, in terms of the latent modeling field. But I think they do. Other studies have looked at like changes in certain sleep disturbances over time, not within like a latent modeling or a clustering approach, and have largely shown what we have. So you've got, say, mixed or a mixed pattern of disturbances generally tend to decrease over time, but you do see, as I mentioned, that insomnia does increase over time. So there's different studies, or like the different methods all kind of converge and show the same pattern that's emerging there, but just in different ways.
Okay, so one last question. Do the switches between discrete patterns cut across gender and race?
Yeah, I mentioned a little bit about the gender before.
Gender.
Race, there was a few, they were relatively small differences. Typically, we saw that, I think, like obviously the ABCD data set is a very diverse and very rich in a whole heap of different ethnicities and races. So we were able to look at that. I think there was, I think if they had individuals identified as being white, they were more likely to be in the higher sleep disturbance classes. But there was no, like, other than that, there was no like specific ethnic or racial kind of patterns that kind of emerged from the data. So the patterns seem to be kind of common across the spectrum of different racial ethnic categories.
All right. Okay. Thank you very much for your very, very interesting. Mr. Pu Xue Mason is a graduate student researcher focusing on microbiocell factories, which have been extensively engineered to produce chemicals, fuels, and materials. He completed a bachelor's degree in chemical engineering from Rensselaer Polytechnic Institute and is currently pursuing his PhD in chemical and biomolecular engineering at the University of Illinois at Urbana-Champaign. Mr. Xue's research recognizes that the metabolic engineering of microbial cell factories remain time-consuming and labor-intensive. And a major reason is the lack of a general platform for large-scale mutagenesis and high-throughput screening. And to address these limitations, he has performed genetic engineering and phenotypic screening by integrating the design, build, test, learn cycle with a biofoundry. And it is our pleasure to have him share his findings with us here today. Mr. Xue, please.
Hello, everyone. My name is Pu Xue Mason from University of Illinois. So in one sentence, I work with robot daily to solve bioengineering questions. I got the inspiration from undergraduate chemical engineering unit operation lab, where each equipment plays its own role and when integrated, fills the ultimate goal. I applied the similar concepts and used the robotic arm to connect each equipment and build a fully automated platform, which can help researchers perform daily repetitive experiments. As a bonus, they can focus on more complicated design tasks. So the scope of iBio5 is to develop automated processes for biological researchers, include metabolic, protein, and cellular engineering. Moreover, it can do some basic cloning work and the DNA assemblies. And then in the future, we want to apply the robot system to help us to do screening of desired phenotypes. And here I listed the available equipments and the modules and its corresponding workflows in the bottom left side. And all this can be connected to each other to perform the multiplexing tasks. And all this has been connected using the momentum software, where the scripts are behind and helps the robot to do the physical liquid handling and do analysis in the downstream end. So what we want to do is by using the automation to do a more rapid design, build, test, learn cycle to fulfill the synthetic biology goal. And overall, we have developed this DBTL cycle and perform tasks using the iBio5 robot to develop a computational design framework and to build the processes handling cells, cell cultures, and even strain validations through our test module and the build module. And in the future, we also want to integrate the learn module to our DBTL cycle, where the robot can help itself to determine what's the next task or next step to do after running several rounds of mutations of the libraries. And in one project, we were working on making plasmids in a large scale to make thousands of plasmids that can be used in synthetic biology. So as scientists in biotechnology, this audience may already have the knowledge that the plasmids are very important in our daily life. And now I just want to give emphasis of why we want to study plasmids. So the construction of plasmid DNA from smaller DNA parts to form complex DNA systems such as metabolic pathways is important for the DBTL cycle in synthetic biology. And the plasmids can be used for delivery of important materials into cells or engineering of various pathways. And some genes can use plasmids as a vehicle to enter the cells for downstream processes, as I shown in this figure. But now the difficulty is that the assembly of these complex plasmid DNAs often requires trials of multiple techniques. So in the end, we can conclude that the construction of plasmids is still time-consuming and labor-intensive. So we want to find better ways to build a lot of plasmids and help them to express in certain cells. So that comes to our strategy used in this project, where we applied the ARGO-based artificial enzymes to do the cutting and the ligation. This was done manually before, and what we want to do is to fully automate this part. So some background knowledge, the PFARGO uses the phosphorylated 16 nucleotides single-stranded DNA guides to cut a single strand of DNA and cleaves the ssDNA target between the 10 and the 12 nucleotide position of its ssDNA guides. And these two precisely designed guides enable cutting of the double-strand DNA with sticky ends of any desired size. So as you can imagine, we can cut the DNA fragments at any locations and then reassemble them to form new plasmids. And this method has been demonstrated successful to assemble up to 18 kilobits of plasmids from 11 fragments in a one-pot reaction. So what we want to do here is to integrate our design, build, test cycle into this artificial enzyme-based method by using our iBio5 system to create an automated workflow for construction of multiple plasmids at one time. So there are four major steps. The first is we want to build a user-friendly web front interface to help us to design the oligos. And the second and third step is to prepare the place for PCR reactions and perform the one-pot digestion, ligation, and the transformation. And in the end, we can confirm our generated plasmids using restriction enzymes and the sequencing. So in details, we want to build this iBioFab as a service to our own campus facility or even to other universities as a collaborator to do the made to order plasmids. So to do that, we already built a web front interface where researchers or customers can design their singular or libraries of plasmids and search our inventories to generate their own plasmids. And once that happened, our iBioFab technician will take the orders from the customers and place the orders through the other company to other companies to help us to generate these primers. Then the robot can do the one-pot plasmid assembly, starts with PCR reaction in a high throughput manner. So here I showed the detailed three modules of our robotic plasmid assembly module with fragment preparation through PCR, the actual assembly, and the confirmation step. In each of these steps, I listed the input that we need and the output of each module. And you can see I want to emphasize here is the human intervention is very low. With our researchers only need to confirm the correct amplification or correct colonies being generated. If not, then we just repeat the modules using the robot. So by doing this, we can build around 100 plasmids within 3 days using this iBioFab system. And in the future, we plan to, as I said before, make this as a service to the campus. So we called the website as a plasmid maker, where the customers or researchers can build their own plasmids. And we are currently also build up our LIMS system, which is the laboratory system that can help us to build a lot of inventories with a lot of templates of plasmids that the customers can just select from our database. And in the end, after the robot, we can generate the correct plasmids and send back to the customers. So in the end, I want to give my acknowledgements to my advisor, Professor Huimin Zhao, and all Zhao group members. And also I want to thank my parents a lot for their support. And here is just a short story that I have been diagnosed with type 1 diabetes during my PhD, but I got the support from my Zhao family and as well as my parents. So they helped me to got over that difficult times. So I really thank the opportunity to study in Huimin Zhao's lab. And I also want to thank the audience for your listening today. Thank you very much. And I will take any questions from here. Thank you.
As well as sharing your personal experiences in your research journey. So we have some questions coming in from the audience. And the first question is, the audience is interested to know what is the cost of your proposed technique compared to traditional methods?
Yeah, that's a very good question. So yeah, indeed, for the traditional cost of generating DNA fragments, it's generally cost like $7 per DNA fragments. And then if we want to assemble them together, say if there are 6 or 8 fragments, so each plasmid may cost around $50. But by using our robotic system, indeed, if we only do several or like 10 or 20 plasmids, that's not worth to use the robotic system. But if we are building a large library such as 100 or 200 or building a lot of plasmids for the company, then this will save a lot of money. So the only cost we have is to getting the primers. Then the downstream parts, all the equipments are involved, and those are standardized processes for every kind of plasmids, and also other projects as well, not only for building plasmids.
I see. Okay. So there is another question. Is there a limit in the number of plasmids you can put together in a single reaction? And how do the robots deal with the differences in the sizes of the plasmids?
Yeah. So so the limitation of how many plasmids really depends on the plate we are using. So currently we are using a 96 well formatted plate. So ideally we can do 94 plasmids at the same time, because we want to leave 2 wells for checking, like putting the control, like positive controls and those things. But we can also always increase the plasmids in one pot to 384 or even 1536 wells. But yeah, as you may know, if we increase the amount of plasmids, there will be more chance to getting unexpected errors. So currently we are, I would say we are limited, or we are focusing on 96 plasmids at the same time.
I see. Okay.
Yeah.
So you also mentioned that you need to ask a company to synthesize primers before generating plasmids. Were you thinking about automating it fully? That is, to start from automated order reading and primer synthesizing?
Yeah, that's a good idea. Like currently, yeah, we are generating the Excels and listed all the things we want to order. Then we send for certain companies such as Twist Bioscience or IDT to synthesize for us, and that will cost money. But in the future, like in universities or other like collaborators, we may start from the scratch just to synthesize the primers by ourselves. Yeah, but that will definitely be a future goal.
I see.
Go, yeah.
I see. All right. So thank you very much for sharing your presentation with us here today. I think that's all the questions we have.
Thank you. Thank you very much.
Thank you very much. Dr. Michelle Nguyen is a research fellow at the Disaster Risk Analytics for Society Lab in Nanyang Technological University. She completed her bachelor's degree in mathematics with first-class honors at Imperial College London, a master's in applied statistics with distinction at the University of Oxford, a PhD in mathematics research back at Imperial College, and then a postdoc after that at the Big Data Institute back at the University of Oxford, before taking up her current position at NTU. In today's talk, she will introduce the classical geostatistical model and highlight the ways it has been applied, adapted, and combined with other statistical techniques to help answer questions in context, ranging from public health to disaster risk. Dr. Noon would also outline ongoing research which uses concepts from information theory to improve model calibration of complex physical models with spatial data. Ladies and gentlemen, please join me in welcoming Dr. Noon.
Hi everyone, my name is Michelle Nguyen, a research fellow at the Asian School of the Environment at Nanyang Technological University in Singapore. My main research interest is spatial temporal statistics, and recently I've been looking at this in the context of environmental and disaster risk modeling. So before I start, I'd like to thank the organizers for this chance to share my research. So for today's talk, it will be centered around the topic of spatial dependence. Is it a boon or bane? So suppose that we have data collected over a study region, like those marked by the crosses on this map here. Then given our observations, how can we estimate what we might observe at an unsampled location? And this brings us to the area of spatial prediction. For today's talk, we'll follow one man's approach to this problem. And this man is Professor Daniel Craig. And he was looking at this problem in the context of gold mining. And the method that he developed was eventually named after him, kriging. So kriging starts by characterizing how different measurements are as a function of their distance apart. And this is expressed in mathematical terms through the construct called a viridogram. This is defined as half the expected squared difference between measurements at two different locations, E of S1 and E of S2. And this quantity is also directly related to perhaps more familiar measures of variation, the variance and the covariance. Specifically, we talk about the spatial covariance because we are interested in how measurements at two locations covary with each other. So once we have modeled the viridogram using our data, we can plug its values into so-called kriging equations to give us the best linear unbiased estimates for the unsampled locations. So this traditional kriging approach is in line with us assuming that the process of interest is a Gaussian process with the corresponding spatial covariance. For such a process, any n observations is assumed to have n-variate normal distribution with a covariance matrix whereby the entries correspond to the assumed spatial covariance function as well as the locations of interest. If we want to include other factors which might affect the spatial variation in our observations, then we typically do this in an additive fashion, and this will give us the classical geostatistical model as shown in this equation here. Here, y corresponds to our observations at a particular location S. X is our vector of covariates. Beta is our perimeter coefficient vector, which tells us how these covariates affect our observations. And E is a mean zero Gaussian process, which tries to capture the residual spatial variation. So one way that we can estimate our parameters of the model is to maximize the likelihood of us observing the data that we have. And this means we use a maximum likelihood estimation method. And once we have done that, we can then use the conditional normal distributions to estimate at the unsampled locations given our observations. So the formula for this classical geostatistical model is very similar to that of a linear model or linear regression, which many of you might have come across. But the key difference here is in the distributional assumption for E. So in a linear model, we assume that E is an independent, identically distributed, normal random variable. However, here we explicitly model the spatial dependence present. So in the figure to the right over here, the blue lines might represent a trend that we see just from the covariates themselves. And under the assumption of independent residuals, then what we might observe might follow something more like the orange line, which varies randomly about this. In the presence of dependence, however, what we see now on the right is regions of sustained overprediction and underprediction. And as we'll see later, this will have impacts. So the classical geostatistical model has been used in various contexts, and it has also been extended to better suit different use cases. So one way that has been done is to change the linear model to a generalized linear one. So in the context of estimating malaria prevalence, this has been done by changing the linear predictor, which is described by x beta plus e, from the real line to the 0 to 1 scale using a link function g. And then we can model our observations, which is typically the number of infected individuals at a particular location, as a binomial distribution in order to estimate its parameters, which is then p, the prevalence. So this approach has been used in spatial epidemiology, and the results, such as that seen on the right, which are maps of prevalence, have been used to track progress and to plan interventions to tackle the disease. Latent variables have also been used to build upon the classical geostatistical model. So a latent variable is a variable which is not directly observed, but it is inferred through other variables which are. So this method has been proven useful in one of the projects that I'm currently working on about estimating earthquake building damage. So conventionally, earthquake engineers model damage by relating it to underlying ground motion intensity measures, such as log of PGA, which is peak ground acceleration. So the higher the intensity measure, the higher the expected damage. And so a latent variable for the propensity for damage might look a bit like Z equals to X beta, as shown here, where X is log PGA and beta is some positive coefficient. However, in practice, in data poor regions, this log PGA is often modeled rather than measured, and studies have shown that there are spatially correlated differences between the model and the measured log PGA. In addition, there is also a widely held but less examined view that there's additional spatial correlation in damage that is not fully attributed to the chosen ground motion intensity measure. So in this project, we are trying to model this phenomena as well as provide some evidence for it. So one way we can do this is to add Gaussian processes for the different factors of the spatial correlation to the latent variable. So for the spatial correlation in log PGA, we can add E of S over here. For the additional spatial correlation in building damage, we can add U of S. And then based on the mean of this latent variable, we can compute probability of exceeding certain damage grades. So in the bottom left plot over here, we have denoted by the vertical lines cutoff values for the different damage grades. And the shaded area here represents the probability of exceeding damage grade 5, because we are looking at the area beyond the 5th cutoff point. So modeling spatial correlation in damage has implications for estimating the risk. And in particular, we show using simulations from earthquake models as well as this damage model that when we account for more sources of spatial correlation, we see higher risk of more extreme portfolio losses, regional portfolio losses. Spatial correlation and modeling spatial dependence has been useful for prediction, but it is a double-edged sword. Studies have also shown that there are underappreciated consequences of neglecting spatial dependence when it is present. Specifically, when we assume that we have independence, we are actually making the assumption that the more data that we gather, the better we can estimate our whatever process that we're interested in by improving its accuracy as well as precision. However, when there's actually dependence, then we have less independent samples than we are assuming, and this would lead us to overstate our confidence and underestimate our standard errors, and could potentially also affect variable selection and lead to model misclassification. Such an issue also arises in environmental model calibration, where, for example, scientists might use spatial surveys, like that shown in the map over here, for volcano ash load, to try to estimate or try to model the process of volcano ash dispersal. And one way that they do this is to optimize loss functions. But these loss functions often implicitly assume independence, such as mean squared error. So one upcoming project that I'm looking at is to develop a weighted calibration procedure to incorporate spatial correlation in model calibration. And this would incorporate ideas from information theory, such as spatial entropy, whereby the information contained in an observation is calculated as a function of its distance from its neighbors, as well as the spatial dependence present. So stay tuned for more of that. To keep in touch, as well as to find out more about my other work, here's a QR code to my personal website. I'd like to take this chance to also thank my collaborators, especially the people at the Disaster Analytics for Society lab at NTU. Thank you for your attention.
Thank you very much, Michelle, for sharing the findings of your research. So we've got some questions coming in from the audience. So first question is, besides earthquake modeling, can you share any other applications potentially for your model over here? Yes.
Yeah, I think it's quite general. So fragility models or damage models have been used for a lot of natural hazards. So you have typhoons, and in that case, instead of peak ground acceleration, you might use like the wind speed, because what might happen there is actually you might be interested in the roofs, whether they get damaged by the wind and so on. And then for floods, you might look at the water depth. And yeah, so I think these models are quite applicable. You just have to adapt them in different situations, and then the spatial correlation could represent something else. Yeah, so it might be representing some ways in which there's spatial differences between your maybe hydraulic model and what you observe, and so on. Yeah.
Right. So a related question, I guess, and one that's probably quite relevant to our current situation, is whether we can employ spatial information between incidents and vaccination centers data sets to map the distribution and prevalence of COVID-19. Yes, so what are your views on that?
Yeah, that is an interesting question. Thank you for that question. Actually, even I would say it is possible. I think I know that people are looking into so-called like catchment areas for health facilities. So I think that is along the same lines. So even when we were looking at malaria, for example, we were looking at where the cases were being reported and where was the nearest health facility, and looking at how people seek treatment as well. So looking at treatment seeking patterns even spatially. So yeah, I think there has been some work there, but it's still very much in the early stages, I think.
Great.
Yeah.
So, so the audience also thinks that, this is a very cool application of GP regression, and they're interested to know whether you're able to apply MCMC methods to capture uncertainty in your infit parameters.
In the infit parameters?
Yes, MCMC methods to capture uncertainty in your infit parameters.
Oh, inferred parameters. Yeah.
Yes.
Yeah, definitely. So there's been a lot of work on Bayesian spatial models, and some people have used MCMC methods. An increasingly popular one is actually an approximation method. It's called INLA, so integrated Laplace nested approximation. So that's also based on very much a Bayesian framework, but also approximation, and a lot of people have been using that. Because one issue when we are dealing with spatial Gaussian processes is really the dimensionality problem. So you have theoretically you would have large covariance matrix that you have to invert and take the determinant off and so on. So yeah, a lot of work has been done in that direction as well.
Okay. So there is one more question. By spatial dependence, they wanted to clarify you mean spatial correlations between local sites, correct? And
yeah.
And how do you define the correlation functions or specify the correlation lengths? Is that from real systems?
Oh yes, yeah. So okay, there are many ways you can go about it. So there are many different spatial correlation functions. I guess the most popular one is right now is the Merton because of its flexibility, and also because it has when you have like a Gaussian process with a Merton covariance, you can link it to stochastic partial differential equations. So there's a nice link there, and you can borrow techniques from the two different disciplines. Yeah, but otherwise, yeah, you can also for other correlation functions link it back to stochastic differential equations, and then some people have done that, I think, in the wave equation, and then you can derive like what kind of correlation functions you should get. Yeah, so
I see. All right. Thank you very much for taking the time to share your research again with us and answering all the audience's questions. Yes, thanks very much, Michelle. Mr. Suteng Chen is a graduate student researcher focusing on the transformation of artificial intelligence for clinical practices by applying deep learning algorithms in intelligent diagnosis and prognosis prediction of patients with malignant tumors. He completed a bachelor's degree in medicine at the Southern Medical University, as well as a master's degree in surgery at Shanghai Jiao Tong University. And Mr. Chen is currently pursuing his doctoral degree in surgery at Shanghai Jiao Tong University. Today, he will be sharing his work proposing novel neural networks for the training and optimization of practical models, which have been verified to perform well in automatic and quick identification of malignant lesions. Ladies and gentlemen, please join me in welcoming Mr. Chen.
Thank you very much. I'm so glad to have the opportunity to take part in the summit. My research interests focus on the transformation of artificial intelligence for clinical practice. So the topic I want to share today is clinical use of deep learning strategy in renal cell carcinoma. Renal cell carcinoma is one of the most aggressive cancer. It represents about 2% to 3% of all cancers. It is also the most common solid lesions within the kidney. As a heterogeneous disease, there are three main types of RCC, and about 70% of this kind of cancer can be categorized as clear cell renal cell carcinoma, which is also called CCRCC. And the preliminary type accounts for about 15%, and about 5% of RCC is familial type. In clinical practice, we found that CCRCC accounts for most metastatic cases and deaths of patients with renal cancer, renal tumor. The accurate diagnosis of CCRCC and if infectious distinguishing with other subtypes is very important. However, there are still challenges there are still difficulties in diagnosis of RCC. The obvious symptoms always lead to delay of hospital presentation, And about the populations of the CT examination, the traditional imaging diagnosis model of renal cancer might be difficult to meet the increased number of cases waiting for diagnosis. So traditional diagnosis by naked eyes of experienced physiologists remains stable, sensitive, and time-consuming. As we can see in 2019, David and his colleagues reported that the physiologist workforce from 2007 and 2017 in the United States. As we can see in the left figure, the number of physicians have increased year by year, which might reflect the increased number of patients. But as we can see, between the same period, the number of active physiologists in the United States decreased by about 18%. This might suggest a shortage of physiologists in the United States, while in China, the shortage of physiologists is also obvious. So we are trying to do something to help to reduce this shortage. Artificial intelligence is the theory and development of computer science, which is able to perform tasks that normally require human intelligence. It is attracts attentions in medical image analysis and molecular drug designation. And in the whole healthcare spaces, AI have proposed to play more and more major roles across a spectrum of application domains, including the diagnosis, the diabetes, the population health management, administration, and the regulation. So AI have also been primarily used in CCRCC, which have focused on the diagnosis, the molecular subtyping, and the survival prediction. However, the limitations of current studies could still be found. For example, some studies are mainly focused on machine learning algorithm, which might suffer from poor robustness and without biological intelligibility. For whole slide imaging of physical expressions, vector information is individual for clinical use. Sample differentiation of RCC is far from enough to be applied in clinical practice. So during the past few years, we have constructed on the use of artificial intelligence in clinical practice. We propose novel neural networks for the training and optimization of practical models. We propose an attention-based weighted supervised learning strategy. As shown in the figure, our classification procedure could be classified into two parts: patch-level feature injections and based on self-supervision learning, and WSI-level feature algorithm based on a deep attention machinism without requirement of manual announcement. This work have covered four classification tasks, including CCRC subtyping, mutation prediction, nuclear grading, and prognosis prediction, which are progressed in 5 branches with prevalence cost anti-loss functions. Firstly, the subtyping results were shown in here. High accuracy of the subtyping symptom was identified in patients caught with LG value of from 97 to 99 plants, which is capable to an experienced physiologist. We also provide interpretability and the visualization of the subtyping task. As we can see, the Kalisa types seems to have more clear functions, while the other type of RTC has more phenotypic course linked by the nuclear cells. Next, we also apply deep learning status to nuclear grading for CCRCC. Generally speaking, tumors with high nuclear grading might be with higher levels of malignancies, but this task is currently performed by experienced physiologists. But now, our AI-based system can help to facilitate this task. Immunotech inhibitor targeting the PD-1 can improve survival of patients with renal cancer. As a current study reported that patients with loss function of PD-1 mutation was associated with higher survival rates. So these patients might benefit more from this kind of anti-PD-1 therapy. So the prediction of mutant status of PD-1 seems to make sense before the therapy. So based on our deep learning strategy, we trained a mutation prediction model for mutation in renal cancer. And this task is difficult because mutation of genes is unlikely to change the phenomenon of a physiological image. So the AI-based network might not find enough useful image features to make an accurate prediction. And finally, the AOC value arrived at about 83% in the training code and 82% in the test code. So this part of task will be deeply investigated in our future work. And finally, we developed and verified a survival prediction model based on the physiological images. Patients with favorable or worse clinical outcomes can be distinguished from each other. KM survival analysis reveals that our deep learning strategy can help to find patients with high survival risk, which might need more follow-up treatment. So with a slight image input, our deep learning strategies can automatically accomplish tumor segmentation, tumor subtyping, nuclear grading, mutation prediction, and survival prediction, which can be presented as an AI-based physiological report. Currently, we hope that our system can help the doctor to finish their daily work more effectively, since the system can provide useful hints in the slide images. Okay, that's my report, and thank you very much.
Right. Thank you very much, Suthen, for sharing the findings of your research. And we have some questions coming in from the audience for you. So first question, the audience is interested to hear you share a bit more details about how your deep learning model was developed. For example, the size of your training data and your testing data. And yes, we'd like to hear you share a bit more details about how that was done.
Okay, thank you very much. Because the present time is limited, so I just primarily introduce my training model. Actually, our prediction model was trained from about 800% from public database, and we prepared about 4 clinical tasks to including the tumor subtyping, the grading, the nuclear grading, the static gene mutation, and survival prediction. And after we training the model and perfect it, we validated it in about 400 patients in our hospital, and our results revealed that the model could be mostly validated in our patients.
Right. I see. Okay. Thank you very much for that. Yeah, so the next question is, I think the audience is interested to know how your model's accuracy compares to traditional machine learning models that I guess have been done in previous works.
That's a good question. Actually, we performed a machine learning model in our previous work, and the accuracy was about for the subtyping of renal cancer. The accuracy was about 85% in our previous published papers, and now we are perform deep learning models in the same task. So and we model accomplished about 99% accuracy in the current work. So we conclude that our current model based on deep learning might perform better than our previous published machine learning based models. Thank you.
Great, I see. Okay, and can you share a little bit more about your future plans moving forward in terms of perhaps bringing this model that you've developed into the clinic? Are there any roadmaps or plans for that to happen?
Oh, thank you. That's a good question. Actually, firstly, we just finished our primary study on this subject, but it's really hard work to transform our current study to clinical practice. maybe we plan to vary out our plan in more medical centers to actually present the economic or medical benefits to the patient. Then maybe we can think about transforming our model to a more commercial use or to some public hospitals to facilitate the production of traditional medical process. Yeah, this is our goals to produce such studies.
Great. So I think this might be the last question for you. So does the model take into account the effect of hypoxia in the tumor microenvironment?
Actually, when we train the model, we have included different kind of renal cancer patients. So maybe some special cases or hard cases have been included in our model in the training process. So I think it will also you can also perform well in coupled with some different kind of hard cases.
Great. I see. All right.
Thank
you
you
very much for taking the time to share your findings today, as well as answering our audience's question. And yes, I think that's all we have for today. Thank you very much for your time.